In this chapter, we're going to learn a lot of fundamental principles about proteins. Proteins in your body or any organism that's a single cell or multi-cell, it's the proteins that do everything. And when I say everything, anything you can think of that happens in a cell or between cells, it's the result of many different types of proteins interacting. It could be a chemical reaction, because I know chemical reactions are the first thing you think about. It could be movement, your muscle movement, your immune system, how it works, the structure that holds your cells together. Anything that happens in your body is the result of proteins. To begin this chapter, we'll look at some basic concepts of proteins, and as we go along, we'll see structures of proteins and how they're related to different functions in your body. And we'll see that the shape of a protein, which is also called its conformation, for most proteins, if that shape changes just slightly, its function changes. And that'll be a guiding principle as we go along from this chapter into upcoming chapters like the enzyme chapter, which are proteins. Proteins are polymers. They're biological polymers found in every organism. And even a simple cell like E. coli produces over 1,000 different types of proteins, different types of polymers. The monomers are amino acids, and there's a set of 20 amino acids that are used to make every protein in every organism, everything that I consider alive, everything that has a cell. We use approximately 23 different types of amino acids to make our proteins, but there's 20 that are common to all organisms. Proteins, they have amine groups and they have carboxylic acid groups. What makes an amino acid different is something called the R group. Here's the basic structure of every amino acid, and again, these are monomers that are used to make polymers. What makes one amino acid different from another is what's here. What's that R group? This R group is called a side chain. These side chains vary a lot with functional groups and polarity. And we'll see a little bit later, the structure of proteins, which is related to its conformation, which is related to its function, depends upon what amino acids and what the R groups are for those amino acids. So here's a carboxylic acid and here's the amine group. We'll see the chemistry in just a little bit, but when the monomers, these amino acids, are used to make the bigger polymers, it's the carboxylic acid and one amino acid. It makes an amide bond with an amine group in another amino acid. So that's some chemistry that we'll discuss and you should remember from your organic chemistry. But we'll see that in a few slides. The two functional groups on an amino acids, excluding whatever the functional group may be on the R group, the carboxylic acid, it's an acid, and it can go through an acid-base reaction and form the carboxylic acid anion. The amine, an amine functional group, it's a base, and it can react and gain a hydrogen cation and form the amine cation. If you remember your discussions about pKa's, and we're going to use those again in this chapter, which functional group form is highest in concentration depends upon the pKa. So an amine, there's an amine functional group, and here's the amine cation functional group. So the amine's a base, and it grabbed a hydrogen cation. There's a pKa for that reaction. And depending upon the pKa and the pH, one of those will be higher in concentration. As far as the carboxylic acid, there's a pKa for the carboxylic acid, and if it reacts and forms the carboxylic acid anion, there's a pKa for that acid, and depending upon the pH and the pKa, one of those will be highest in concentration. In this chapter, and for amino acids, I'm going to give you those pKa values. But if we look at an amino acid, here are three forms of the amino acid. They're all the same amino acid, but those functional groups, whether it's the amine or the amine cation, what form is that amino acid in? It depends upon the pH. Is it the carboxylic acid or is it in the anion form? Which one is highest in concentration depends upon the pH. All amino acids are a little bit different because their R groups are different, but in general, the pKa's for all the amino acids, they range from 1.8 to 2.6. So what range, are they higher, are they 1.8 or are they 2.6 for the carboxylic acid? It depends upon the R group. For the amine group, or the amine cation, which is actually the acid, the pKa's range from 8.8 to 10.6. So the amine cation, that's the acid, it can lose an H plus and form the amine group. Which pKa it has depends upon the R group. If we look at the information packet, and if we look on page 3, here's information about the equilibrium of the amino acid at different pHs. And instead of giving you a range of pKa's, you're given something here. If the pH is less than 2.6, this form of the amino acid will be highest in concentration. If the pH is greater than 10.6, this form of the amino acid would be highest in concentration. And when we write amino acids, we always write the one that's highest in concentration. If the pH is between 2.6 and 10.6, the middle one is highest in concentration. All three forms are in equilibrium. It doesn't matter what pH you're at, all three would exist. But when we write the structure, if we're going to use an amino acid in a chemical reaction, and I give you the pH, you need to use this chart to figure out what form you're going to write your amino acid in. So is it going to have the amine cation and the carboxylic acid, or maybe it's the carboxylic acid anion? So let's go back to our lecture notes. So all three of these molecules are in equilibrium. So I'm drawing arrows like that. There's some equilibrium. If the pH is less than 2.6, then this concentration is highest in equilibrium. If the pH is greater than, and we'll put than, 10.6, then that one's highest in concentration. The middle one, if the pH is between 2.6 and 10.6, then the middle one is highest in concentration. So in your stomach, where the pH can range from 1 to 3, well, when it's below 2.6, this concentration, the one on the left, is the highest in concentration. So if I said, the stomach, and let's just say it's the pH equal to 1, then this form would be highest in concentration. In your body, you never reach a pH that's 10.6. The highest, the most basic pH in your body is in your intestinal tract, where the pH may be as high as 8. Most of your cellular fluids, so your blood within your cells, the pH is really, really close to 7.4. And it's this form that would be highest in concentration at a pH of 7.4. And the tables of amino acids that are in our information packet, they're all drawn in this middle form because that's the one that's highest in concentration. One note, when we look at those tables, the carboxylic acid anion, which is shown here more expanded, is written in the condensed form. So here's the condensed form of a carboxylic acid. I'll draw it condensed and expanded throughout. A couple of notes, the R groups, some of the R groups, a few of them, a handful of them, also have acid-base functional groups, and those R groups can have a charge. Most of the amino acids in most R groups do not, but certain ones do. And we'll learn that the ones that are charged can lead to some structural conformations of proteins. Another note about amino acids and proteins in general, they can have a net charge. So the amine groups, the amine cations, create a positive charge, and the carboxylic acid anions create a negative charge. If we look at the first one, so first one from left to right, the charge of this, the net charge, is positive 1. There's a cation functional group in that structure. If we look at the middle one, there's a positive and a negative. The net charge is 0. If we look at the amino acid when the pH is greater than 10.6, the net charge is -1. Shown on this page is a listing of some of the amino acids. A complete listing is shown in your information packet. So this shows all 20 amino acids, all 20 common amino acids. They're divided up based upon the structure and the functional groups in the R groups. The very top one, if we look here, are R groups that are considered nonpolar. The next one is polar. And the bottom one are R groups that have acid-base functional groups. In our lecture notes, the first slide shows the nonpolar R groups. So if we look at the R groups, which are also called the side chains, all the basic structures of the amino acids are the same except for the R groups. There's the amine cation in a condensed form, and there's the carboxylic acid anion. This table is assuming that we're at a pH of 7.4 in your cellular fluids. But what are the R groups? That's an R group for that one. And if you're an amino acid and you have an R group that's just a hydrogen atom, that amino acid is called glycine. There's 3-letter abbreviations and single-letter abbreviations that are used for the amino acids. And if you're glycine, that 3-letter abbreviation is GLY. And the single letter abbreviation is G. When we use abbreviations, we'll be using the 3-letter abbreviations. Notice all these R groups have nonpolar bonds. So there's an R group, there's an R group, and here's another R group. And they all have different names. Note, amino acids are very, very polar. They're charged. They're cations and anions. They're not nonpolar. The designation here is for the R groups. And we'll see later it's these R groups and their composition that leads to structure in a protein. On this slide is the middle section of amino acids from our table, and these amino acids have side chains that are considered neutral and polar. And if we look at these, there's kind of a common theme with most of these except for one. All of these amino acids, the side chains, there's an OH, OH, there's some nitrogens, oxygens. These side chains, most of them in here, all the ones that I have circled, those side chains can hydrogen bond. And we'll see a little bit later that hydrogen bonding, those attractions, lead to structure within proteins. But again, amino acid is used to make proteins, and we'll see these R groups that are part of the protein structure. All of the side chains except for one can hydrogen bond, and that's the one with the thiol group. If you remember here thiol chemistry from organic chemistry, a thiol group, 2 thiol groups can react to form a disulfide bond. We'll see that chemistry in just a little bit, but forming disulfide bonds is an important structural component for certain proteins. This last slide of the table, it shows the bottom part of the amino acid table, and these side chains, these are called basic polar or acidic polar. These are the side chains that have acid-base functional groups, and these are the ones that can chemically react and make positive or negative charges. So the ones on the left, these are basic ones, and again, we're showing all the amino acids shown here are at a pH of 7, and if you're a amine functional group and you're at a pH of 7.4, the highest concentration is the amine cation. And we can see that concept of amine cation structure looking at our information packet for this part that we looked at a little bit earlier. So here's the amine. I know it's part of an amino acid and it's not the R group, but still kind of says the same thing. If the pH is less than 10.6, then the amine functional group is in the cation form. And it doesn't matter, going left from 10.6, that direction, it's in the cation form. The carboxylic acid, if we're at a pH less than 2.6, whether it's part of this structure or part of the R group, it's in the carboxylic acid form. If the pH is greater than 2.6, then the carboxylic acid is in the anion form. And again, it doesn't matter if we're talking about that functional group being part of the R group or not. it's going to be in that particular form. So again, this whole table assumes we're at a pH of 7.4 and the mean is in the cation form. The carboxylic acid, which those are the two amino acids that have this in their R chain on the right, aspartate, aspartic acid, and glutamate, glutamic acid, they're both carboxylic acid, but they're in the carboxylic acid anion form. And note, one of them has 3 carbons in the R group, and one of them has 2. They look pretty close. Just make sure you're real careful when you're trying to figure out what R groups they are when we look at other examples later on. But as far as properties of these R groups, these are the R groups that can have charge, and it's these bottom ones. And you can have charge interactions, electrostatic interactions, and we'll see that later. Some proteins have a lot of electrostatic interactions, attractions and repulsions. And also remember, whether something's charged or not, whether it's the anion form or the cation form, it depends upon pH. pH changes can affect whether the R groups are charged or not, which can actually affect structure, but we'll see that a little bit later too. When amino acids bond together to make bigger structures, they're called peptides or polypeptides. If there's enough amino acids bonded together, it's called a protein, and we'll define how many amino acids it takes a little bit later. On this page, we're going to look at the chemistry that occurs. If 2 amino acids bond together, it's called a dipeptide. The chemistry that occurs, it occurs between the carboxylic acid and the amine group, and we form an amide structure. However, when it's a pH of 7.4, the carboxylic acid is not in its neutral form, it's in the anion form. And the amine, it's in its cation form. So this chemical reaction that we're showing here, it has those functional groups, the amine and the carboxylic acid, in their appropriate structures based upon being at a pH of 7.4. So as far as the functional groups that are reacting, it's the carboxylic acid anion plus the amine cation. We form an amide functional group plus water. This chemical reaction could occur in a test tube in water. This amine cation group could collide with that carboxylic acid anion group, and we could form an amide functional group. However, molecules can move around, and this amine cation could collide with that carboxylic acid anion, and you could form an amide group. If that happened, you actually would form an isomer. The isomer it would form because things are a little different. The R groups aren't the same. In your body, though, it's not an easy one-step reaction like this. And in your body, it's very specific what amino acid gets bonded to what when you make polypeptides. It's a multi-step process, and we'll talk about it at the end of the chapter. But just to kind of give you a general overview, the proteins that are made are based upon the genes in your DNA. And again, we'll talk about this at the end of this chapter. Your DNA is used to make RNA molecules. And your DNA, it's in the nucleus. The RNA is synthesized in the nucleus. The RNA then goes into the cytosol, and it's in there where the RNA is used to make your proteins. And it's a multi-step process. It's pretty complicated. There's different types of RNAs that we'll show you, and it requires chemical energy, it requires ATP. It's pretty complicated. But for right now, we'll look at what functional groups are reacting and what's being formed. And for the product that's shown on the right, on the product side, it's this functional group reacting with that functional group. So when this happens, when this reaction happens, this nitrogen, there's a collision, and it forms a bond with that carbon. That carbon-oxygen single bond breaks, and 2 of the NH bonds break. Those 3 together form H2O. It's this bond right here that's forming, that's linking the 2 amino acids together. And what's formed on the right, this is a dipeptide. It's made, di meaning 2, it's made from 2 amino acids. It's no longer amino acids, but it has the they're formed from the amino acids, and we say there's residues from the amino acids. But if we kind of put a circle around things, this portion all the way to that nitrogen, that came from this amino acid. The other portion came from this amino acid. For this chemical reaction, there's a few things you should be able to point out and label. The bond that forms between the two amino acids to make the dipeptide, it's called a peptide linkage. It's called the peptide bond. You should be able to point an arrow at that bond that forms between the two amino acids. So it's somewhere in there. Also, use your information packet, look at these two amino acids, write the 3-letter abbreviations for each amino acid. So why don't you go ahead and pause the video and do those 3 things. Unpause the video when you're ready to check your answer. So looking at the R groups, this R group, it's an H, and if we went to our information packet, what only has an H? Well, we pointed out earlier, it's glycine. So right there. The other amino acid, it has 1, 2, 3, 4 carbons in its structure. There's its R group. They're all nonpolar bonds, so it's up top. What has 4 carbons? Hopefully you didn't pick valine. So LEU, leucine, there's 4 carbons. Looks a little different, but just the confirmation, how we drew it is a little bit different. Some of you may have looked at isoleucine, but the first carbon coming off of our amino acid is CH2. So it's leucine. As far as the peptide bond, it's this bond that formed right there. That's this bond right here that's forming. That's called the peptide bond. And if there's 2 amino acids, there's 1 peptide bond. We could bond more than 2 amino acids, and that's what we're going to see on the next page. If there's 3 amino acids that are connected together to make a polypeptide, it's called a tripeptide. And shown on this page is our same dipeptide from the previous page. And here's another amino acid. Different R group has a different name. All proteins, all polypeptides are made within your cellular fluids. And again, it's a complicated process. It uses the RNA molecules and enzymes. And again, we'll see that a little bit later, but we'll just accept that it works somehow. But when this reaction happens, let's assume it's this functional group and that functional group react to form a tripeptide. Why don't you go ahead and pause the video and complete this reaction. Unpause the video when you're ready to check your answer. So shown here is the tripeptide that forms. And again, we're assuming it's this amine cation reacting with that carboxylic acid anion. And we're also assuming that the pH is 7.4. So remember, one of the products, it's always water, and down here is our tripeptide. Shown here, that's the peptide bond that formed. So that's that bond that was forming between this nitrogen and that carbon. What's interesting for all tripeptides that form, this portion up here that's in red, that whole portion, that's the same from one tripeptide to another. What makes a tripeptide different are the R groups that are used to make the tripeptide. There's a few vocabulary terms we can add since we understand that concept a little bit. The portion in red on this tripeptide, this is called the backbone. And this is a definition that we're going to use to make other definitions later. So it's good to understand that. The R groups, they're still called the side chain. So these are the there's 3 side chains for this tripeptide. Every tripeptide would have 3 side chains. A dipeptide would have 2. Another terminology for peptides that are formed, the amino acid, that portion of the tripeptide, they're called residues. There's 3 amino acids to make the tripeptide. There's 3 residues. And we could put a circle around all 3 of them. And when you do the circle, you go from one nitrogen atom and stop right before you come to the next one. So if we go from left to right, here's a nitrogen. We'll go all the way to here. That's a residue. I mean, it's part of the tripeptide, but it's an amino acid residue. Next, we'll go from this nitrogen and stop right before that nitrogen. So if we put a circle around that residue, whoops, got to get the whole R group. There's the middle residue. And the right one, that's the last residue. Later in this chapter, we'll be circling residues. You'll be given a longer polypeptide, and you'll be circling residues and indicating interactions between residues. So we'll use this again later. Another designation are the end residues. The end residue that has the amino acid cation functional group is called the N-terminal residue, which means the one on the left, this residue right there, is the N-terminal residue. The one that has the carboxylic acid anion residue is called the C residue, carboxylic acid C, and that would be the one on the right that contains the carboxylic acid. Shown here is the general structure of a dipeptide. And every dipeptide would look exactly like this at a pH of 7.4. What would make each dipeptide different would be the R groups, which would come from the particular amino acids. This portion here that I have circled, that's the backbone. Why don't you pause the video and write the general structure of a tripeptide. Just remember as you do this, a tripeptide would have 3 amino acid residues, which means there should be 3 R groups coming off of your general form of a tripeptide. Go ahead and pause the video and unpause when you're ready to check your answer. Here's the general structure of a tripeptide. On this structure, the green color, all those atoms in green, that's part of the backbone of this bigger molecule. The red colors, these are the side chains, and for a tripeptide with 3 amino acid residues, there would be 3 R groups. But again, every tripeptide would have the same backbone. What makes the tripeptides different is the carbon atoms that are part of the side chains. On this page, we're going to learn how to name a polypeptide. And don't worry, it's not nearly as complicated as that IUPAC nomenclature you learned for carbon-based molecules. When you name a polypeptide, you indicate what are the amino acids that are part of that polypeptide, and you have to give the order of the amino acids. When you start labeling the amino acid residues, you start on the N-terminal side. And the way we're going to do it, we're going to use the 3-letter abbreviations to indicate the amino acid residue. So if we look at our information packet, if you remember, there's 3-letter abbreviations and there's single-letter abbreviations. All of my examples, like for glycine, it's GLY, we're going to use the 3-letter abbreviations and not the letter G. The polypeptides that we're going to name, there may be just a handful of amino acids that are part of that structure. So here's a polypeptide that we're going to name. This is the N-terminal side, so we're going to name this polypeptide based upon this amino acid residue, and we'll be looking at that R group. In fact, this is the same tripeptide that we saw earlier. Here's the next amino acid, and here being right there, and we'll name this amino acid residue based upon that R group. And here's the third R group. If we remember what it's formed from, glycine, leucine, aniline, so we have GLY, LEU, and ALA for the 3 amino acid residues. So this one is GLY, that residue, LEU, and ALA. So as far as naming this, put those 3-letter abbreviations together, GLY, and you can put a dash line if you want, you don't have to, LEU, and ALA. So that's naming this tripeptide. Note, this is a tripeptide, and this is a particular tripeptide with these 3 amino acids. You could actually form 5 other isomers, including this one. So 6 isomers from 3 amino acids. And we're going to see how we can do this in just a second. But in this particular molecule, the glycine and its residue is the one that's next to nitrogen. Well, what if leucines was first and it was next to nitrogen? That would make a different molecule. Or what if this R group was the middle one and not the end one that's on the C terminal side? That would make a different one. So this is, that's one isomer. If you still had glycine at the N-terminal side, but ALA was next and then LEU was next, which means this CH3 was part of the middle residue, that's a different molecule. That's a different isomer. What if this R group was the one that was next to the nitrogen and it was on the N-terminal side? So this could be the first one, and maybe glycine was next and then ALA was last again. That's a different isomer. No matter how you rotate any of these three molecules around, they're different molecules. A fourth way, we would still have leucine on the N-terminal side, but ALA next and GLY on the C-terminal side. Here's 4 different isomers if you have 3 amino acids, but there's 6. The 6th way, so we'll do 5. What if ALA, and we'll make that an A, what if that was first? And then maybe glycine next, and then leucine last. And a 6th way, and the last way, ALA first, and leucine next, and then glycine on the C-terminal side. For 3 amino acids, you could have 6 isomers. And don't worry, for the exam, I won't have this type of problem with more than 3 amino acids. If there was 4 amino acids, and you thought of all the different orders, there would be more than 6 isomers. If it was a dipeptide, you could have 2 isomers. But for our exam, if you had a question like this, 3 amino acids. And you'll see this type of problem given to you in the laboratory for practice. Earlier, we learned that an amine plus a carboxylic acid formed an amide group plus water. Well, to break that amide group apart, all you need is water. That's basically doing the reverse reaction. And that's what we're showing down here. Here's a dipeptide. There's our amide group. There's the peptide bond that formed. To break that apart, all you need is that water molecule to collide right there, and it breaks that dipeptide back into the 2 amino acids. And that amide group would reform the carboxylic acid and the amine group. This reaction, this hydrolysis reaction, can occur with water or the hydronium ion. And that hydronium ion, it can collide with that peptide bond, break that, and reform the amine and the carboxylic acid. Now, what form do those function groups have once they become products over there depends upon pH. So the top one, that's at a pretty low pH. Is the amine in the amine form or the amine cation form? Is the carboxylic acid in the carboxylic acid form or the anion form? And here's the reaction at a pH of 7.4. So the top one would be the reaction that could occur in your stomach where you have a pretty low pH. The bottom one is where the reaction would happen throughout most of your body, in the blood or in your cellular fluids. Why don't you go ahead and pause the video and complete this reaction. So there's 2 products, 2 amino acids. Unpause the video when you're ready to check your answer. So since we're at a pH of 1, the amino acids should be in this form. The amine should be the amine cation, and the carboxylic acid should be in its neutral form. So that amide functional group in that dipeptide reacts with the hydronium ion, and we form the carboxylic acid and the amine cation for the functional groups. So this collision breaks that peptide bond right there, and we have glycine and leucine. For both of them, it's the amine cation in the amino acid, and the carboxylic acid is in its neutral form. Next, I want you to pause the video and complete the bottom reaction. This reaction is happening at a pH of 7.4, and it's water that collides with the amide group and breaks that bond. You're still making the 2 amino acids, but they'll be in the form that they should be at pH 7.4. So pause the video, unpause the video when you're ready to check your answer. At a pH of 7.4, well, 7.4 is between 2.6 and 10.6. The amino acids that you write should be in that form. So the amine cation and the carboxylic acid anion. The amide group, when it reacts with water at this particular pH, you form the carboxylic acid anions and the amine cations. And that's what we see in our 2 amino acids. Just a few notes about these 2 reactions. So this is breaking down a polypeptide. You can have hydrolysis reactions of polypeptides. If you just put a polypeptide into water, that reaction is incredibly slow. I mean, you can take some meat that has lots of protein in it, put it into water, and nothing really changes. In order for the rate of this reaction to occur dramatically, to break down proteins, you have to have enzymes. And again, we'll talk about enzymes in the next chapter, but that's needed to increase that rate of reaction, breaking down peptide linkages. In your stomach, though, where the pH is really, really low, there's relatively a large amount of hydronium ions. And you can, under acidic conditions, have a relatively high rate of reaction with hydronium ions. Remember, pH is related to the hydronium ion concentration. And remember, rates of reactions, as the concentration increases, the rate of the reaction increases. And if you remember, pH is related to hydronium ion concentration. So I'm pretty sure you could fill in this statement that I quickly wrote. The rate of this reaction increases or decreases as pH decreases. Why don't you go ahead and pause the video and write increases or decreases, and unpause the video when you're ready to check your answer. The rate of this reaction increases as pH decreases, and that's because the hydronium ion concentration is increasing as pH decreases. And just one note for abbreviations. So there's the hydronium ion. Putting brackets like this, that means concentration. So rate of reaction increases as pH decreases, hydronium ion concentration increases. If you've ever worked with food and you've tenderized meat, what you're really doing is breaking down the structural proteins by adding chemicals. And you can do this by adding enzymes that'll catalyze this reaction, or you could add acid to your meat and break down those structural formulas. And what's often used is acetic acid that's in vinegar. In your body, for chemical reactions, as far as the breakdown of peptides, it only dramatically occurs at a high rate in your stomach. If you remember, you always have the hydronium ion in any cellular fluids, but when the pH is 7.4, that hydronium ion concentration is so low that the rate of that reaction is too low to be considered substantial for your metabolism, and your body uses enzymes. But just note, in any aqueous solution, you always have the hydronium ion. And that means, it may be a slow rate, but it means if you do put a protein into water, it'll eventually break apart into the amino acids because there are some hydronium ions. The last chemistry that you need to remember from your organic chemistry class that we'll be using in this chapter is thiol chemistry. And your organic chemistry instructor would be so happy if you could remember the reaction that happens between 2 thiol groups. If you remember this reaction from that class, a thiol functional group plus a thiol functional group plus some oxidizing agents, and there's different types of oxidizing agents. When these chemicals are put together, you make a disulfide. And depending upon what the oxidizing agent is, those 2 hydrogens that are part of the thiol group, they could go into solution and form 2 hydrogen cations, or maybe bond to the oxidizing agents. In your body, what are some oxidizing agents? We've learned about a couple oxidizing agents in the previous chapter. What were they? They were coenzymes, and we had acronyms. One of them was FAD, and the other one was NAD+. Those are oxidizing agents. You have other types in your body, but those are 2 oxidizing agents that are in your body. The place where we'll see thiol groups in thiol chemistry in protein molecules and protein synthesis is in one particular amino acid, and it's cysteine. That R group has a thiol group. If you have two of those R groups come together, you can have a reaction in which you make a disulfide functional group. Why don't you go ahead and pause the video, and here's your two amino acids. These two thiol groups are going to react. Why don't you complete the reaction assuming that FAD is the reactant? So go ahead and pause the video. Unpause the video when you're ready to check your answer. So when this reaction happens, a bond is created between the two sulfur atoms. That sulfur hydrogen bond in both of them breaks. So here's the bond that's created between the two sulfur atoms. This is called a disulfide linkage, this bond that forms. This functional group in protein chemistry is also called a disulfide bridge. The FAD, since we said that's our reactant, on the product side, it would be FADH2. Those two hydrogens from the two thiol groups bond to the FAD molecule. If this was NAD reacting, then it would be NADH plus a hydrogen ion. In the next few slides, we'll look at example polypeptides in which disulfide bridges, disulfide linkages are formed, probably polypeptides whose names you recognize. One of them is very, very common. Note that if you wanted to break a disulfide linkage, if you wanted to break that disulfide bond, so say we had some sort of general structure, let me draw a line to separate. Say we had R, S, and an R. If you wanted to break that bond and reform two molecules, you need a reducing agent. FAD and NAD+, that's an oxidizing agent. To break that bond in the way your body does it, what molecules does it use? It uses NADH or FADH2. And using those molecules, you can reform the thiol groups. And depending upon which one you use, let's just say it was FADH2, you would form FAD. Okay, one other type of chemistry that was given in organic chemistry around thiol groups, you need an oxidizing agent for this reaction to happen. There's some oxidizing agents that change the final structure around a little bit. And if you remember, lead 2+ cation or mercury 2+ cation can be the oxidizing agent in a thiol reaction. So what if we had two thiol groups? So RSH plus RSH. And the oxidizing agent, let's say it was lead the cation. So Pb2+. There's something that forms. Why don't you go ahead and pause the video and write the generic product. Unpause the video when you're ready to check your answer. If lead or mercury cations, if they're present when these disulfide bridges are being created, you still get that connection. You still make a bridge, but instead of being sulfur to sulfur, you get an extra atom in that bridge. And you may say, well, should that really matter if that happens when we're making disulfide bridges in our proteins? Would making it just a little bit longer matter? And we'll see in a little bit, it could make a big difference. Changing the structure slightly just by one atom in a big protein molecule could make a big difference on whether it actually works or not. If it works, meaning that protein has some sort of function. One note, this reaction can happen with lead 2+ or mercury 2+. And it's this reaction with these cations, these metal cations, it's harmful when the disulfide bridge is being synthesized. These are oxidizing agents. They're not going to break apart a bridge that's already been synthesized. So if you've already synthesized a protein and it has disulfide bridges, linkages, adding lead and mercury, those cations, it's not going to hurt that protein. It's when proteins are being synthesized that it can be very dangerous. Shown on this page are two example polypeptides in which there's a disulfide bridge within its structure. Both of these are hormones. One is called oxytocin, and the other one is vasopressin. Both of them have 9 amino acid residues, and this drawing here, the circles represent the amino acid residues. So it's a little different than what you've seen, 1 all the way through 9. For both of them, there's a disulfide bridge, a linkage that's created between the R groups of amino acid number 1 and amino acid number 6. Both of those are cysteine amino acid residues. What makes the structure and ultimately the function of these two polypeptides, what makes them different, are 2 amino acids at 3 and the 8th position. So the R groups are slightly different there, but having 2 small changes in the R groups dramatically changes the functions of both of these molecules. And again, both of these molecules are hormones. Hormones are produced in one cell, they're secreted, and then they eventually attach to other cells. There's receptor proteins on these other cells that are super specific to hormone molecules. And these hormone molecules have to fit just right into those receptor proteins. And having 2 amino acids that are different in the structure makes it so that oxytocin can't fit into vasopressin's receptor molecules and vice versa. They both are produced in the same cell. Their target cells are slightly different. And what I wrote there, both are hormones and both are produced in the pituitary gland. The target cells for vasopressin, and vasopressin is also called the antidiuretic hormone. Some of the target cells are the kidneys, and this is released when you're dehydrated. This hormone vasopressin also targets blood cells and brain and other cells. Usually hormones target multiple cells. Oxytocin, it's also produced in the pituitary gland, and it targets cells like the uterus. Okay, one note about information that I've given you here, I don't expect you for this particular slide to have to memorize information about these 2 hormones. Again, look at the extra problems, and that'll be a great guide for you to help yourself prepare for that exam. If there's information from a slide that I expect you to know for the exam that seems like there's a lot of information about a particular protein, I'll tell you that. Just like I told you to have all of those structures of those 7 chemicals in the previous chapter, be able to label those. There's some things in this chapter that I expect you to memorize, but when we get to those slides, I'll make sure I point it out. Here's another example of a polypeptide that contains disulfide linkages, and it's a protein that you've heard of, or probably heard of. It's called insulin. Insulin has 51 amino acid residues. What's a little different about insulin compared to some of our examples previously, one, it's bigger, but 2, here's 1 chain of amino acids, and here's a second chain of amino acids. So there's chain 1 and chain 2. These 2 chains are chemically bonded together via thiol groups reacting to make disulfide linkages. And that's what's shown here and there. There's also a disulfide linkage within chain number 1 between R groups. And we see that example right there. So these are 2 polypeptide chains that come together to make insulin. Shown down here is the actual 3-dimensional shape of insulin. Looks a lot different than these little circles. Insulin is also a hormone, just like the oxytocin on the previous page. And its shape is just right to fit into some sort of protein receptor. There's lots of different target cells for insulin. This 3-dimensional shape is also called its conformation. So what I wrote here, the 3-dimensional shape, which is also called its conformation, the shape, the biological function depends upon this shape. And small changes in its shape can have a big influence on its function. For example, if insulin was being synthesized and there was lead or mercury that was present, when these disulfide bridges were being made, adding one mercury atom in that bridge or one lead atom in that bridge can have a relatively major change in the shape, meaning it may not fit into that protein receptor. You may still synthesize this protein, insulin, but it's not really insulin anymore. It may not work with that lead or mercury that could be part of its structure. Another thing to note, this three-dimensional shape, it's conformation of a protein. This shape starts to take place as the peptide is being synthesized. We'll learn in a little more detail later on in this chapter how polypeptides are synthesized based upon your gene expression. But trust me for right now, the synthesis of polypeptides, it's extremely consistent, meaning the order of amino acids, how they're bonded together, it's the same most of the time. Which means if the structure starts to take place, the shape starts to take place as it's being synthesized, and that's extremely consistent, we get a consistent shape of the protein too. Polypeptides are sometimes called proteins, and sometimes they're just called peptides. And here's the definition for you. If there's 50 or more amino acid residues, it's called a protein. So, what example did we see that's considered a protein? Well, that's insulin. Insulin has 51 amino acid residues. And it doesn't matter that there's two chains, both of those chains together make one polypeptide protein. If there's 49 or less amino acid residues, it's just called a peptide. So oxytocin, that's considered a peptide. Some of the smallest proteins, well, that would be insulin with only 51 residues. Insulin, there being insulin, the molecular weight of insulin is around 6,000 grams per mole. Some of the bigger proteins that are found in your body can be millions of grams per mole. Here's a larger one that's close, myosin is close to 1,000,000 grams per mole, and it has 6,100 amino acid residues. All proteins are synthesized within cells. Some are secreted into the blood to serve some function like insulin. Some are always kept within the cell as long as the cell is living. However, when cells die, the components of the cells are released into the blood, and because of that, and because cells are always constantly dying, every cell in your body, there's a certain death rate. No matter what, your muscle cells, there's a certain death rate, even your neurological cells, even your heart cells. But luckily, you remake new cells too. But when they die, they release their components to their blood. All proteins have a certain concentration in your blood, and we've tested normal and healthy people, and there's certain normal concentration ranges. Our hearts, there's a certain normal death rate. And the release of these proteins from your heart into your blood, there's a normal concentration range. If for some reason this concentration, you went and had a blood sample and this concentration was way above normal, that could indicate a problem. What if you had a heart attack and all of a sudden you had a large rate of death of your heart cells, and it would release all those components? Those proteins that are in your heart cell would have a higher concentration in your blood. There's been lots of tests that have been developed, which we call assays, that look for protein concentrations in the blood, and often we look for proteins that are very specific to certain cells. Like there's certain proteins that are only expressed, only synthesized in your heart cell. And we know what a normal concentration range is for those heart proteins. If that concentration is above normal, then it tells us that you probably had a heart attack. It's a super, super specific medical test. And we have certain proteins that are specific for your kidneys, your liver, any type of cell that you can think of. There's certain specific proteins, assays that have been developed for those proteins. And if you think you have a thyroid problem, well, there's specific protein for that. The doctors know what a normal range should be. And if you ever looked at a medical chart, if you've seen a biological sample sent in and tested, they look at protein concentrations, and there's always normal ranges. We'll look at this more in the next chapter. And in fact, one of your laboratories coming up is called Diagnosing Disease. You're going to be testing some serum samples, which are blood samples, to see if that person, and you're testing two different people, so persons, if one of them has a problem or if both of them are healthy. Okay, I just wrote a quick paragraph on what I just said, and let me read it in case you have a hard time seeing my handwriting. And when a cell dies, so I should add an S there, their contents are released to the blood. Different proteins have specific concentration ranges in your blood. Again, there's some proteins that are secreted and have a function in your blood, but what we're looking at are proteins that are used within the cell and they become part of the blood when a cell dies. Normal concentration ranges have been established for proteins found only in one type of cell, meaning we've taken normal and healthy people, taken their blood samples, and analyzed what's the normal concentration, and it's a range because everyone's a little different. If you took a person that came to the hospital and they thought they had a heart attack, and if their heart protein was found, if that concentration was above the range, then it indicates a problem and they probably had a heart attack because you had an increase in heart cell death. However, if that heart protein concentration in the blood was at a normal range, it could mean maybe they have a hernia or something. On these next few slides, we're going to learn about the protein structure. What type of interactions give proteins their shape, their conformation? So for insulin, what's going on to give it this three-dimensional shape? There's four main aspects to a protein structure. There's the primary structure, which we'll talk about on this page. There's also the secondary structure, which we'll start talking about on this slide. There's the tertiary structure that we'll talk about on this slide. And there could be a quaternary structure that we'll start to discuss on this slide. So the primary structure. This is the order of the amino acids and how they're linked together. And again, it's pretty complicated. It goes from your DNA to your RNA, and we'll see that a little bit later. But your DNA, and there's things within the DNA that are called genes, and there's some sort of order in there. Those are used to create RNA molecules, and they're produced in a certain order of monomers. These RNA molecules are then used to make polypeptides. And your body is super, super good at producing the proteins just right. Your body does it like 99.99% of the time. Just to kind of give you an idea of what are the possible isomers that could happen if you had insulin and there's 51 residues, how many isomers could you have? And forming just from the amino acids, from the monomers, there's 1 times 10 to the 66 isomers by just changing the order of the amino acid residues. That's quite a bit. Your body is super good at making the correct order almost every time. Luckily though, even if the body, maybe you get one amino acid wrong in oxytocin, what's kind of nice is even though you get one amino acid wrong some of the time, that protein usually doesn't work. So you make a bad protein. But if 999 proteins are good out of 1,000, your body's going to function. Another thing that's pretty unique about your body, and we'll see this later when we look over your metabolism of proteins, but amino acids, again, they're used to make polypeptides. You have certain chemicals in your body that are called enzymes, and you make some per cell whose sole purpose is to break down any protein, any polypeptide that you make, and break them back down into amino acids. And every protein has something that we call half-life. How long do they last? Why would your body do something like this? Well, one reason, sometimes you make bad polypeptides, and this is a way of getting rid of the bad one and restarting over with a pool of amino acids. And two, you're full of water. You have hydronium ions in there. And Even though it's a really low concentration of hydronium ion in your blood or your cellular fluids at a pH of 7, those proteins, if there's water, they're going to react with the hydronium ion once in a while. So even if you made 999 good ones, pretty soon one of those amide peptide bonds is going to break. Proteins don't last forever in a water-based solution, which is you. So there's lots of strategies in synthesis of protein and breaking them back down into amino acids. But let's get back to the primary structure. A lot of the proteins in your body, and you have thousands of proteins, most of them, or close to half, have been sequenced, meaning we know the order of amino acids. And as we mentioned up above, the sequence of amino acids in a protein and a polypeptide, it's determined by your DNA. It's determined by the genes in DNA, which is said to be genetically determined. Missing one amino acid or getting one amino acid out of order or something can make it so that that particular protein polypeptide doesn't work. We're going to see the structure of hemoglobin in a few slides, but sickle cell anemia, it's a genetic disease in which two of the amino acids in hemoglobin, two wrong ones are used. And the hemoglobin and the red blood cell that it's part of doesn't work as well, and capillaries end up being clogged. It's an awful, awful disease. But just getting two wrong amino acids can have a dramatic effect on function. The next structures we're going to talk about, secondary, tertiary, and quaternary, remember all of these start to form as the protein chains are being synthesized. Secondary structures, which can be abbreviated as a 2 with a naught, this interaction, it's actually an attraction. It's hydrogen bonding, and it's very, very specific. So there's some definitions we're going to be giving you. It's hydrogen bonding between the amide groups on the backbone on the same chain. Hydrogen bonding between amide groups on the backbone on the same chain. So I repeated it twice. But some of these words in here came from definitions that we gave earlier. So remember the R groups, those are the side chains. All these other atoms that are here, these are atoms that are part of the backbone. It's also between the amide groups, not the amine group, not the carboxylic acid group, but the amide groups that are on the polypeptide. This is what an amide group would, it's part of the chain, so there'd be an R and an R. So remember what hydrogen bonding is. Hydrogen bonding is between polar bonds. And the definition of hydrogen bonding, it's the partial positive H in an OH bond or an NH bond. That partial positive H is attracted to a partial negative O or N in another polar bond. So in this amide group, what do we have? We have a partial negative oxygen and a partial positive carbon. The nitrogen-hydrogen bond, partial negative nitrogen, partial positive hydrogen. Remember to think of a polar bond as a magnet. So there's the north and there's the south. And this one, it's a north and a south. Okay, when you have hydrogen bonding, those magnets need to get next to each other. It's not hydrogen bonding between these two bonds. It'd be this amide group and some amide group further away. So here's another amide group, and these would be connected because they're on the same chain. So remember, insulin has two chains. This has to be hydrogen bonding between amide groups that are on the same chain. So it would have to be hydrogen bonding between amide groups on that same chain or this same chain. It can't be hydrogen bonding between two different chains. So hydrogen bonding, even you have the word bonding, it's an attraction. And it's an attraction between this magnet and that magnet. The hydrogen bonding that occurs in protein structures, just because of the geometry and how the functional groups can get next to each other, it's the attraction between the partial positive H in the NH bond, so this one, and I'm going to draw a dashed line for that, and it's attracted to the partial negative oxygen in that CO double bond. So this north is attracted to that south. That's hydrogen bonding. And this leads to the structure in proteins. there's two different types of secondary hydrogen bonding structures that can form. On this page, we're looking at one that's called an alpha helix. This is kind of like that helical structure that you saw in starch when you took organic chemistry. And you get some sort of coiled structure. This occurs between amide groups that are 4 amino acid residues away, and it's always 4 away. So somehow you have this polypeptide chain. This chain can start coiling and wrapping back on itself, and if it's 4 residues away, you can have this alpha helix type of structure. So let's look at this generic polypeptide down here, and it keeps going that way and that way. But let's put a circle around all the residues, and end to end makes a residue. So from there to there, that's residue 1, and we could keep going to 3, 4, 5, and 6. And we'll just write, we have the R1, R2, but we'll just write 1, 2, 3, 4, 5, 6. So you could have alpha helix type of structure between amide groups that are 4 residues away. So you could go from 1 to 5, the amide groups that are between 1 and 5, or you could show alpha helix hydrogen bonding between residues 2 and 6. And when you do it, it's the carbonyl, the oxygen there in one amide group, and the H in the NH bond in another amide group. So if we were going to draw a dashed line, if I went from residue 1, so we could go from that oxygen and go all the way over to residue 5. So I'm making a dashed line, dashed line, dashed line, over to residue 5. And we need to go down to the NH bond that's in residue 5, which is right there. So that's an example of an alpha helix hydrogen bonding interaction. You could have went from this H and that NH bond in residue 6 and that O in residue 2. Note that in order to have this helical structure, the chain kind of has to wrap around and fold back just a little bit. Think of a protein, a polypeptide, like a Christmas tree strand. And each light represents an amino acid residue. In order to have hydrogen bonding, those functional groups have to be able to get close to each other. So you need some wrapping around. They can't be too close. You can't be just one residue away. That's too close for bending. Also, these R groups that are here, they're usually, if you have alpha helical type of structure, those R groups are usually small, so something like glycine, and they're usually not charged. So you wouldn't see this if there was a lot of acid-base functional groups in there that were charged. Not every protein has alpha helix structure, and some proteins, not everywhere in the backbone do you have this structure. It just depends upon the sequence of amino acids. There's lots of ways to illustrate alpha helical structure, and that's what we're trying to show here. You could use a more complicated one using spheres for all the various atoms. That gets kind of complicated. This ribbon here, that represents the backbone, and we'll see that in a lot of the different illustrations we're going to show. So the ribbon represents the backbone, and to me that's a lot easier to see than all these different balls that represent the various atoms. Okay, so this is alpha helix. On the next page we're going to see something that's called beta pleated, and it's also a secondary structure. Beta pleated structures are also secondary structures, and it's still hydrogen bonding between amide groups that are on the same chain. But for beta pleated, the hydrogen bonding between the amide groups are more than 4 residues away. And in fact, they may be hundreds of residues away. But you still get this overlapping, and amide groups on different parts of the chain come in contact with each other, and you get this hydrogen bonding interaction. So this illustration down here, it may seem like this is 3 different polypeptides, but I'm making a loop there. It just means that it's one big long chain, one big polypeptide. So maybe we should write that off to the side. And when you get this beta pleated structure, what does it look like if we looked at it with light scattering? Well, it looks like a pleat, and it's baited, so it's called baited pleated. And just like with alpha helical structures, you don't have baited pleated structures in every protein. And some proteins you have more and some you have none. So it just depends upon the amino acid sequence. Shown on this page is a diagram that tries to illustrate the two different types of secondary structures. And remember, this ribbon shape, that represents the backbone. Why don't you go ahead and pause the video and label the two secondary structures. Unpause the video when you're ready to check your answer. So in this diagram, there's only one spot where you see a baited pleated structure, and one site where you see an alpha helix. We'll see examples of real proteins where you see multiple sites of both. When you label baited pleated, you can write the word baited pleated or use the Greek symbol beta. Same thing for alpha, you can use the alpha symbol alpha. Either one works perfectly. But just remember, this is hydrogen bonding between amide groups that are on the same chain. The next type of structure we're going to analyze that you find within polypeptides is called the tertiary structure. And if you want to abbreviate it with a 3 and a naught, that's perfectly acceptable. But tertiary structure, here's another definition. It's the result of interactions between the side chains, those R groups of the amino acids, not the backbone, but the side chains, that are on the same chain. So we'll write that in. So it's interactions between R groups that are on the same chain. So this is just a definition. So if you have a chain, you can have secondary structures, which are, they're interactions, but it's hydrogen bonding between amide groups that are part of the backbone. Tertiary structures are interactions between functional groups that are on the same chain, but it's the R groups. So it's a little different. One's called secondary, one's called tertiary. They're both interactions that can lead to a three-dimensional structure. Secondary interactions, it's just hydrogen bonding interactions. For tertiary, however, there's 4 different possible types of interactions that could occur. One of them is also hydrogen bonding. A second one is called salt bridges. On the next page, we see the other 2. The third one is called disulfide bridges, and the last one is called hydrophobic interactions. Again, these are between R groups. We can use our amino acid table and we can look at the definitions, the listings of the amino acids, and certain amino acids groupings does one of these 4. So if there was hydrogen bonding, if you had this type of interaction, you need R groups that have the correct type of bonds that could do hydrogen bonding. We need some polar bonds. We probably need an OH bond or an NH bond. Hydrogen bonding, if we look in our table, what group of amino acids could do hydrogen bonding? It's the neutral polar amino acids that could do hydrogen bonding. Well, at least all of them but one. Any one of these listed down below, there's an OH bond, OH, OH. Remember, you need an OH bond or an NH bond in order to have hydrogen bonding. Tryptophan can do that. Here's amide group. All of those, if you had 2 of these R groups next to each other, you would have hydrogen bonding, except for cysteine. That SH, that's a nonpolar bond. It doesn't have a partial negative or partial positive. It's like a piece of plastic. So, written here, 2 R groups from amino acids in the neutral polar grouping, except for cysteine. And remember, you need polar bonds, and never ever use a CH bond, that's a nonpolar bond, or an SH bond to show hydrogen bonding. So we'll put a big X there. And remember, hydrogen bonding is like having 2 magnets, a north and a south, next to each other. It's an attraction. And you need magnets. And CH and SH, even though there's hydrogen in there, they're not magnets. Okay, salt bridges. These are electrostatic interactions, and they could be positive or negative. It's the attraction between ions. A salt bridge is an ion-ion interaction. So if we went to our table, R groups that are ions, that's the basic polar side chains or the acidic polar side chains. So you could have a side chain in R group that's positive and a side group that's negative, and you have this electrostatic interaction, attraction. One note though, this is called a salt bridge, and if you say salt bridge, you'd think, well, it's an attraction, and that's true. That attraction between these R groups helps hold the protein in a certain shape, but even though it's not implied here with the name, repulsions of R groups have an equal effect on the shape of the protein. So we said ion-ion interaction, it really can be electrostatic attractions and repulsions. So let's say you had an aspartate side chain next to a glutamate side chain. They're both negative, that's repulsion. And that repulsion would keep those protein chains, those portions away from each other, and it leads to structure. So salt bridge, you can just call it a salt bridge, but it's really repulsion or attractions. I'm not super particular on how you give names. If you want to say electrostatic interactions, that's perfectly acceptable too. Or ion-ion interactions, that also works with me. But these are two possible interactions that could give rise to a tertiary structure of the protein. But again, the protein, in order to have salt bridges, it needs those acid and base side chains. Not all proteins have acid and base side chains, but these are possible interactions that could occur within a protein. And let's talk about the other two on the next page. The next tertiary structure on our list is disulfide bridges, and this chemistry that you remember from organic chemistry and a few slides ago, it's thiol chemistry, and there's only one amino acid that has a thiol group, and that's cysteine. So if you have a polypeptide chain and you have these R groups, from one amino acid residue with this thiol group and another one, and you have the enzyme, you can create a disulfide linkage. The last tertiary interaction is called hydrophobic interactions. These are London forces, dispersion forces. This occurs between R groups that have nonpolar bonds. And if we went to our table of amino acids, that would be between neutral nonpolar side chains. So nonpolar bonds, nonpolar bonds, nonpolar bonds. So like I mentioned, hydrophobic interactions, this is a protein chemistry term. If you want to use terms that you learned in Chemistry 121, you can call number 4 dispersion forces or London forces. Any of those work perfectly fine for me. On the next page, let's go ahead and practice labeling interactions given a protein. Okay, here's a diagram of a protein. This is the ribbon, and this is trying to be part of an R group. It's not showing the whole R groups. The name of the R groups, the 3-letter abbreviations, they're given there. What I want you to do is pause the video. There's 4 different tertiary interactions. This is just one chain, but label all 4 interactions with the labeling we had from the previous page. Unpause the video when you're ready to check your answer. So the top one right there, that's a disulfide linkage. This one is a salt bridge, attraction between a cation and an anion. Here's 2 nonpolar ring structures, so that's a hydrophobic interaction. And here's an interaction trying to show hydrogen bonding. One last note about these interactions. Just like secondary interactions, these tertiary interactions lead to the shape, the conformation of the protein. Of the 4, though, one of them is a lot stronger than the other one. The one that's a bond, a covalent bond, is much stronger. The other ones are just attractions between R groups. It's like an attraction between 2 magnets. It's the disulfide linkage that's really, really strong. The other 3 are just attractions, and they're weaker attractions. And the reason I'm bringing this up is eventually we're going to learn what can you do, what kind of conditions can you alter to affect the shape of a protein. Well, if you can interfere with these attractions, no matter what type it is, you're going to change the shape of a protein. If you took out a 2x4 on a wall, you're going to change the shape of a wall. There's things that you can do that can alter the shape of the protein, and as we'll learn as we keep going on and on with this chapter and the next chapter, shape conformation is definitely related to the function of the protein. But for right now, what I want you to write down, and let me read what I just wrote. Hydrogen bonding, cell bridges, and hydrophobic interactions are similar in strength. And I also put in there under cellular conditions. So under the mixture environment in your cellular fluids, which is a pretty complex solution. A disulfide bridge, which is a covalent bond, is much stronger. And in order to break that, you need some sort of reactant, like a reducing agent, to break that bond. So some of you that have had general chemistry may be saying, hydrogen bonding, I thought that was super strong, and a hydrophobic interaction, London dispersion forces, was a little bit weaker. And yes, if you're not in that aqueous mixture of your cellular fluids. Why would that be? Well, you have an attraction if these are right next to each other. It's kind of like if you had two pieces of wood, maybe here's my wood, and here's another piece of wood. And if you put maybe some glue, which I'll put in red there, if you put those next to each other and pressed them together, there would be some stickiness, and they would stick to each other. And there's an attraction between the glue and all three of those. Well, what if you put something in between, like a piece of paper? That paper would stick to the glue and prevent this piece of wood from sticking to the glue. That can happen between R groups. There's things in your body that can prevent these interactions. If you had some sort of molecule in here that could hydrogen bond, and it could hydrogen bond to that R group, well, it can prevent the other R group, which is over here, from hydrogen bonding. Kind of the same thing as this glue and piece of paper. Same thing here. This is a cation-anion interaction. Well, if you put some other cation here and it was making an attraction to that anion, it can prevent that one from being attracted. In fact, something like water, H2O, remember water can hydrogen bond. Water's moving around constantly. It can move by, hydrogen bond, prevent this one for a little bit, the two R groups from hydrogen bonding. But remember, these are attractions. Eventually that water can move away. The protein shape that you get is related to how often are these groups being prevented from being attracted to each other, and how often are they attracted to each other. Proteins, peptides, they're not stationary, stagnant, really solid pieces of material. They're vibrating, moving, but they take on some sort of average conformation. And this average structure depends upon average cellular conditions. You can change things compared to your normal cellular conditions, and that can affect the protein structure. There's molecules that you can add to a protein mixture and interfere with hydrogen bonding or salt bridges and affect the protein structure or hydrophobic interactions. We're going to learn in a little bit things that you can do externally that can affect the structure of a protein, which can affect the shape, which can affect the function. The fourth structure, the last structure we're going to look at, these structures are called quaternary structures, and you can draw a 4 with a knot. Quaternary interactions, there's a lot of different types, and it can be the same types that we saw in the tertiary structure. So it can be electrostatic interactions, it could be hydrogen bonding, hydrophobic interactions, disulfide linkages. But the difference between this and tertiary structures is it's the same sort of interactions, but it's between two different chains, or between chains. So we'll complete that statement. Between chains. So primary, that's the sequence of amino acids. Secondary, it's hydrogen bonding between the amide groups on the same chain on the backbone. Tertiary interactions are interactions between R groups that are on the same chain. Quaternary interactions are any interaction between two different polypeptide chains. Some proteins like insulin, there's 2 chains. Proteins like fibrous proteins have at least 3 chains. So let's look at this example on this page. As we're looking at the formation, here's the formation, the primary structure, the sequence of amino acids. And note one thing up here, polypeptide chains, they're also called subunits, although I never say that, but they are. Okay, as the protein is being synthesized, as the chain is being synthesized, you start getting some secondary structures. And also, as it's being synthesized within that chain, you start getting the tertiary structures that arise as those R groups get next to each other. And this is one chain. It's one subunit. Shown down here, if you can see it, there's 4 different chains held together by quaternary interactions. So that's supposed to be 1, 2, that one was the 3rd chain, and there's the 4th chain. So this is 4 chains held together by quaternary interactions. And these quaternary interactions, they could be bonds, they could be just interactions, attractions like hydrogen bonding. But anytime you see 2 chains in an interaction, it's always quaternary. So let's practice this concept. Let's go back to the lecture slide that had the insulin 3-dimensional structure. So this slide. Remember, there's 3 disulfide bridges. So there's one there, there's one there, and there's one there. Why don't you go ahead and label those disulfide bridges, labeling by either calling them primary, secondary, tertiary, or quaternary. When you go ahead and pause the video, label the 3 of them, they're only one type. Unpause the video when you're ready to check your answer. Any interaction between 2 chains is quaternary. So that's an interaction between 2 chains. That disulfide bridge is an interaction between 2 chains. This one is an interaction between R groups that are on the same chain, so that's tertiary. You can also go to your study guide with all those practice problems and look at what's presented. Here's a problem. You're supposed to label interactions between R groups, between these 2 different chains. This is one polypeptide chain, this is another. Even though it doesn't ask it in this problem, that interaction between those 2 chains would be quaternary. Even though it says between the R groups, it's not tertiary. Tertiary would be between R groups that are on the same chain. Any interaction between 2 different chains, no matter what it is and what it's between, is always quaternary. Shown on this page is another example polypeptide. It's hemoglobin. Hemoglobin is a protein whose function is to carry oxygen from your lungs to other cells. It's found within your red blood cells in your vascular system. There's about 1 million of these per red blood cell. Hemoglobin is made from 4 different chains, and those 4 chains are held together by hydrophobic forces. So that's the quaternary structure. I expect you for the exam to know that the quaternary structure of hemoglobin is hydrophobic, and we'll assume for this exam only hydrophobic. You know there's going to be a test question based upon you having that knowledge. If you also look at hemoglobin structure that's shown here, there's probably something that looks familiar and something that looks unfamiliar. What looks familiar is the alpha helic type of structure that you see throughout. What's a little bit unfamiliar is this structure that we see located 1 per chain. This is something that's called a heme group, and there's different types of heme molecules. What's true about all heme molecules is there's an iron cation in the center. The main function, as we said earlier, of hemoglobin is to carry oxygen from the lungs to other cells. The heme, that iron that's at the heme, is the part of the hemoglobin that oxygen attaches to. And if you remember the Lewis dot structure for oxygen, so O2, it has some lone pairs. Those lone pairs, those electrons make a bond with an iron. It's a weak bond to the iron. Weak enough that eventually there's a collision that knocks them away, and then the oxygen can diffuse within the cell. For hemoglobin, it has a certain average conformation within the red blood cell. These heme groups are oriented for the most part so that the iron is accessible to the oxygen that's going to diffuse in. If you have a conformation change, and if that iron portion in the heme molecule was more pointed towards some sort of backbone that made it inaccessible for the oxygen to get to that iron, it could make it hard for the oxygen to make a bond. The hemoglobin actually changes shape, changes conformation as it flows through your vascular system. In a few pages, we're going to state what type of molecule whose concentration changes affects the shape of the hemoglobin, which we'll state in a little bit actually affects the function of the hemoglobin. But we'll talk about that in just a little bit. We'll come back to this slide in just a little bit. On the next few slides, we're going to look at conditions that can change, that can have an effect on the shape of the protein, on its conformation. These conditions can be a physical condition or a chemical condition. These changes in conditions, these changes affect the structures, the attractions, the bonds that give the protein its structure. So all those various attractions that we talked about, like hydrogen bonding, salt bridges, and all the chemical bonds, the amide bonds, the disulfide linkages that form, if you do anything that affects those attractions or bonds, then you're going to change the shape of the protein. So remember what we said earlier, the function of a protein, and there's lots of different function, it depends upon its conformation, which is also called the shape. If you have a shape change, if you have a conformation change, you may think that that change in shape, that change in function would be a negative effect. It could be a positive effect. So what I wrote, shape change could be positive, meaning that protein works better, or it could be a negative shape change, meaning the protein doesn't work as well. Proteins, as far as their function and how well they work, it's also called activity. Function is also called activity. Something like hemoglobin, if its function is increasing, it means that it's working better. It means its activity is increasing. It means it's doing a better job at transporting oxygen, which could mean maybe those heme molecules, the iron portion, is oriented so the oxygen can easily bind to it. Now let's look at conditions that you can change that can affect protein conformation. Shown in this diagram are the tertiary interactions that occur within a protein. One of them is a salt bridge. If somehow you could change something that could make it so you didn't have this attraction that gives the protein its shape, the protein structure could be altered. What's a variable you could change? pH. Remember, the ions, they're formed from an acid-base reaction. Whether they're charged or not depends upon pH. So if there's salt bridges in a protein and you change pH, that conformation of that protein is going to change. So I'm going to make a list, and here's number 1, pH changes. These changes affect the salt bridges. The charge of the side chains of the acid-base R groups depends upon pH. So it affects the electrostatic attractions and repulsions that may exist in a protein. Not every protein has a salt bridge, so pH changes only affect proteins that have a salt bridge. But if you're told a protein has a salt bridge, if the pH changes, the shape definitely changes. The only thing is, you're not quite sure if that pH change is a positive effect or a negative effect. You need to be given more information. We'll learn that certain proteins, there's an optimum pH that it works best at. But that optimum pH needs to be given to you. There's certain proteins in your stomach whose optimum pH is close to 2. There's some proteins in your digestive tract where their optimum pHs are close to 8. You need to be given that information. Okay, let's look at number 2 on our list. And let's also think about a variable that could interfere with salt bridges. So the first one was pH. The other one I talked about earlier when we were describing salt bridges. Remember, this is an attraction, if we look at the attraction aspect of it, between a cation and an anion. What if, and your body has a lot of these, you have solvated ions that are free to move around. And if you get a solvated ion like a cation and it gets between the two R groups that are charged, it can prevent the two from interacting. The higher the concentration of the dissolved salt, the more likely it would be between those two R groups. Changing salt concentration can change the attraction between the salt bridges. Salt concentration affects the salt bridges. So what I wrote here, changing salt concentration, remember those brackets mean concentration, affects the salt bridges. Your body, the cellular fluids in your body, there's a high salt concentration. Your concentration of salt in your cellular fluids is close to ocean water. Your proteins that have salt bridges take on a certain shape based upon having a certain concentration of salts. Going higher or lower of your normal standard salt concentration in your cellular fluids can affect the shape of certain proteins. And again, just like pH changes, you're not sure if that shape change is a positive or a negative effect. You would have to be given like an optimum salt concentration that the protein works best at. Okay, let's look at another environmental condition that can change. Interaction gives structure to a protein could be hydrogen bonding. So if there's secondary hydrogen bonding interactions or these tertiary interactions, if you could do something that could disrupt that attraction, you can change the conformation of a protein. And if you remember what we said earlier when we described this interaction, if you had a molecule that could hydrogen bond and it hydrogen bonded to one of the groups, it could prevent the other one from hydrogen bonding. So this next thing, number 3, changing the concentrations of molecules that can hydrogen bond. So changing the concentration of molecules that can hydrogen bond affects hydrogen bonding structures within the protein. You'll see a lot of practice problems where I describe the protein and I describe what are the interactions that give it its conformation, its structure, and then I may say this environmental condition changes, what's going to be affected. So there's lots of molecules that can hydrogen bond, something like glucose, that's lots of alcohol groups, changing its concentration. Say you had a test tube full of proteins, if you change the glucose concentration and that protein had hydrogen bonding structures, its conformation would change. Okay, let's go to the next slide and do 4. Another structure, another interaction that could be present in a protein could be a hydrophobic interaction. If you remember, these are dispersion attractions. They're London forces. And here's an example of that type of interaction. If you had a molecule in there that had a lot of dispersion forces that was attracted to this R group, it could prevent it from being attracted to the other R group, meaning it could interfere with that sort of structure, that attraction. The way it's phrased for protein chemistry, changing the concentration of these type of molecules, it's said to be changing in concentration of carbon-based solvents. So changing the carbon-based solvent concentration affects the hydrophobic structures. Carbon-based solvents are hydrophobic chemicals. Hydrophobic solvents affect the hydrophobic structures. Okay, what exactly is a carbon-based solvent? And there's a few definitions. And here's one that I'm going to give to you. It's based upon the number of carbon atoms per oxygen atom. And the general rule is for carbon-based solvents, there's 4 or more carbons per 1 oxygen. That's the ratio. And a good example would be diethyl ether. And its formula is C4H10O. That's a carbon-based solvent. Also, there's some other common molecules that are considered carbon-based solvents, and we're not going to use the formula to figure it out, but soaps and detergents are considered carbon-based solvents. So in general, carbon-based solvents, these are nonpolar hydrophobic molecules. So alkanes would be another example of a carbon-based solvent. So something like octane, if you saw the formula C8H18, which is octane, that's a carbon-based solvent. The next variable that we're going to discuss that affects the shape of the protein, that interferes with structures in the protein, is temperature. Temperature, when you have temperature changes, you're changing the velocity, the kinetic energy of molecules. If you remember from your general chemistry, if temperature decreases, then the average kinetic energy of a molecule decreases. If it increases, and it being temperature, then the molecules are moving faster. For a protein, if you increase temperature, it means that the protein, that big structure, is vibrating with more energy. This vibrational energy can break the attractions that are part of the protein. Not covalent bonds, but attractions. So what are the attractions that are in the protein? Well, salt bridges, that's an attraction. Hydrogen bonding, that's an attraction. Hydrophobic structures, that's an attraction. Temperature changes, it's not enough to break covalent bonds like a disulfide linkage, or all those amide bonds, those peptide bonds that are within the ribbon structure. If you took protein, like a piece of meat, and you boiled it in water, that's not breaking the structural proteins. You can go at 100 degrees Celsius, that's not breaking the covalent bonds. But it certainly could be enough energy so that the hydrogen bonding attractions and the hydrophobic and the salt bridges, that's enough vibrational energy to break those attractions. So what I wrote, temperature changes affect salt bridges, hydrogen bonding, and hydrophobic structures, not covalent bonds. There's actually lots of organisms that live close to the boiling point of water, and their proteins work just well at those higher temperatures. If that boiling water temperature was enough to break amide bonds, peptide bonds, then you'd never have a protein that functions at that high of a temperature. Okay, one last note, the temperature changes that occur, they have to be above some sort of minimum temperature. And that minimum temperature is 10 degrees Celsius. Note, this doesn't mean that you have to have a temperature change of 10 degrees Celsius. It just means that there needs to be a temperature change, and at some point the temperature is above 10 degrees Celsius. So a temperature change from 12 degrees Celsius to 14 degrees Celsius will change the conformation of a protein, or a change from 7 degrees Celsius to 25 degrees Celsius, that would cause a conformational change. But if you change temperature, say you started at 7 degrees Celsius and you went up to 9 degrees Celsius, or if you went from 7 degrees Celsius to 1 degree Celsius, those changes are all below 10 degrees Celsius, and there would be no conformational change. So, why would changing the temperature below this minimum amount not affect the shape of the protein? Well, when you're going to lower temperatures, you're lowering the kinetic energy. And below this 10 degrees Celsius, the kinetic energy changes, and the kinetic energy itself isn't enough to overcome these conformational structures that result from those attractions. The analogy I like to use is breaking a pencil. You could ask a 12-year-old, and they could probably break that pencil. That's like breaking an attraction. If you asked a 3-year-old, they could probably break that pencil, break that attraction. But if you go down to a 2-year-old, they probably couldn't. And even if you went down to a 1-year-old, they probably couldn't. So decreasing the age, there's some sort of minimum age at which there's not enough change of force in the two people to break that pencil. There's a minimum temperature. If the changes are all below that minimum temperature, which is 10 degrees Celsius, that's not going to affect the structure of the protein. In fact, if you've ever worked in the medical field, where are biological samples stored? They're stored in freezers that are really, really low in temperature. Going to lower temperatures doesn't disrupt the proteins and kill that organism. If you've been in microbiology, to kill an organism, well, you put the samples at a high temperature, 125 degrees Celsius. Higher temperatures can disrupt the proteins. Lower temperatures, low enough, below 10 degrees Celsius, changes below that do not. Okay, one last thing to write down on this slide, and I'll read it to you. Note, any change to the primary, secondary, tertiary, or quaternary structures in a protein will change the shape, function, and activity of the protein. And again, this could be a positive or a negative activity change. You have to be given some additional information. For example, you have some proteins in your body whose optimum temperature, the temperature they work best at, is close to 30 degrees Celsius. We'll see in the laboratory when we look at the activity of some proteins, some of them work best around 50 degrees Celsius. It just depends upon the protein. Make sure you go to your study guide and look at all those great practice problems to see how this information that you just learned about will be tested on the exam. So here are just some example ones straight out of the study guide. In the first part, you're given information about a protein, you're given some environmental conditions, and you're told that one of those variables changes. And in here, the temperature changed. When you answer the question, you need to state the names of the structures that were disrupted. So like hydrogen bonding, salt bridges. You also need to state whether there was a primary, secondary, or tertiary structure that was altered. So given information about the protein, you should be able to say something like that. So remember, primary, that's the sequence of amino acids. To disrupt that, you need to be able to disrupt a peptide bond, which you're not with temperature. Secondary structures, remember secondary structures, that's hydrogen bonding between the amide groups that are on the backbone on the same chain. Tertiary structures, those are those four interactions that occur between side chains, R groups, that are on the same chain. Quaternary structures, this was a 2-chain or 3-chain protein, there could be quaternary structures. It's a single chain. There's no quaternary structures. You'll see a little bit later with these type of problems, there could be quaternary interactions that were disrupted. But you'll see those a little bit later as you practice these practice problems. Before we move on to a new slide, I want to go backwards and talk about the conformational change of hemoglobin as it flows through your vascular system in your red blood cells. Carbon dioxide is a waste molecule produced by all your cells in your body. It reacts with water and forms carbonic acid. As your blood flows towards your lungs, the carbon dioxide concentration is at its highest. When it flows away from your lungs, the lungs release a lot of the carbon dioxide that's in your vascular system. When the blood goes past your lungs, the carbon dioxide and the carbonic acid concentrations are much lower. Carbonic acid is an acid and it affects pH. So this means you have slight pH changes before your lungs and after your lungs in your vascular system. When carbon dioxide concentration is relatively higher, it's a higher concentration of the acid, it's more acidic and pH would be lower. Point is, we get pH changes, slight pH changes in your vascular system. The hemoglobin within its tertiary structure has salt bridges. And remember, the pH affects whether the charged R groups are charged or not. And you get a change in the structure of hemoglobin as the pH changes. And what's been found experimentally, as carbon dioxide concentration changes, the shape of hemoglobin changes, and as that carbon dioxide concentration increases, hemoglobin's function increases. Activity increases. What does that mean? It means the shape changes slightly as carbon dioxide concentration increases, and those heme molecules, maybe they're more oriented to make it easier for oxygen to be carried or to collide with it. The function of hemoglobin is to carry oxygen. So the activity, the function, remember the same thing. At high carbon dioxide concentrations, where you're getting close to the lungs, the hemoglobin, its structure changes, so it makes it easier for oxygen to attach and make a covalent bond. It's pretty amazing. The structure of proteins change as environmental conditions change. And environmental conditions, what does that mean in your body and for a chemist? It's talking about the concentrations of molecules. The concentration of molecules that are around your proteins, your working proteins in your body, they affect how well that protein works. And what we're going to learn as we go through this quarter, molecules that are produced in the chemistry in your body, those molecules and their concentrations have a big influence on how well the proteins inside your body, how well they work. Some molecules can make the protein work better, some can make it work worse. Carbon dioxide makes hemoglobin work better. On the previous two slides, we learned about some of the environmental conditions that can change, that can alter the shape and the function of a protein. On this page, we're going to learn about extreme conditions, environmental conditions, in which the protein doesn't work anymore. When a protein, when its shape has changed so much compared from its working shape, and it can't ever work again, that protein is said to be denatured. So denatured means the polypeptide has lost all of its biological function. Remember, function is related, it's also called activity. And its function within a cell cannot be restored. So we need to add an E to B. In your body, in an organism, if a protein isn't going to work, if it's not working anymore, there's nothing that your cell can do. You don't have mechanisms to make that protein work again. And the reason I'm stating that, because in the laboratory, you can denature a protein and then renature it. And some classes that discuss proteins, when they use the word denatured, they also include the laboratory experiments. But in your body, if for some reason a protein in your body becomes denatured, it's not going to work again. So what are some extreme conditions that could cause a protein to be denatured? And number one could be too high of a temperature. What would be too high of a temperature and what's it doing to the protein? So remember, you have a protein and there's usually attractions that hold the protein and give its shape. So here we have a protein, maybe its whole shape is some sort of alpha helical structure. When you increase temperature, you increase the vibrational energy of the protein, and that vibrational energy, when it's enough to overcome those attractive hydrogen bonding forces that are giving it its structure, when that happens, then that protein can become more extended and take on kind of a shape like that. If you said, hey, protein, go back to that same working shape, that's most likely not going to occur. And the analogy I like to use is our Christmas tree strand. Remember a Christmas tree strand, that's like our protein. Each bulb is an amino acid residue. Let's say that Christmas tree strand's in a ball, like the ball shape it's in when you get it out of the Christmas tree box after being stored for a season. That ball, that shape, that's the working shape of a protein. And that shape, remember, arises as the protein is being synthesized. You know, when you try and wrap those lights around your tree, you usually get your Christmas tree strand all strung out. So being strung out like this shape. If you took this Christmas tree strand and you started throwing it up in the air and said, come back to that same ball shape that you were before, it's not going to happen. There's an extremely low probability that that would happen. This is what temperature is doing. It's taking some globular shape, some shape conformation of a protein. Temperature can make it extended, break all those attractions. When those attractions start to reform, you just don't get the same shape and it doesn't work anymore. Too high of a temperature, what's too high of a temperature? Most proteins denature above 85 degrees Celsius. However, you have some proteins that denature around 106 degrees Fahrenheit, which is around 42 degrees Celsius. This is the reason why you die if you have a fever for too long. Most people most adults would die if their fever was 107 degrees Celsius for a day. At that temperature, there's a few proteins that become denatured. Most proteins don't, but there's a few that do, which means there's certain proteins that are part of some pathway, some mechanism that don't work anymore. But it only takes one not to work to shut down a pathway. And a few do at that temperature, and denatured means they're not going to work anymore at that temperature at a fever of 107 degrees Celsius. It doesn't happen instantaneously, but when most of them are denatured, that pathway shuts down and that could kill a cell. But also know you're going to be working with an enzyme that you produce, and you're going to be working with it in the laboratory, and you're going to find its temperature that it works best at, and it's going to be closer to 50 degrees Celsius. There's lots of enzymes that work above body temperature. Okay, what's another extreme condition that could denature a protein? Any variable, any condition that we explained before that can affect the shape of a protein, an extreme condition for that could cause it to be denatured. So remember, salt concentration can change the shape of a protein if there's salt bridges within the protein. So too high of a salt concentration can denature a protein. And exactly what's too high? Usually above 10% weight per volume of the salt. So above 10% weight over volume. Too high of a salt concentrations, remember salt concentrations can prevent a salt bridge and an attraction, but even though we didn't really talk about a lot, too low of a salt concentration can denature a protein. So if you took the purified water from the laboratory and you put a protein into it, if there's salt bridges in that protein, it's probably going to be denatured. So remember what I said about salt bridges. You can have cation and anion R groups and you have an attraction. And bridges implies it's always an attraction, but repulsions that are part of the R groups also give structure to the protein. You can have an R group that's positive and another R group that's positive, and that repulsion prevents those R groups from getting next to each other. That actually leads to structure. And to pure water, there's too much repulsion. The fact that proteins denature at too high of a salt concentration is why certain foods are preserved in a high salt concentration, like jerky. Also remember, there's only one denaturing temperature, and it's the extreme high temperature. We store biological samples at lower temperatures. Proteins are not denatured at lower temperatures. Going to lower temperatures, that doesn't break the attractions that's causing the denaturing process to occur at higher temperatures. One note as we leave this page and go to the next one, I'll list a few other denaturing conditions. Just because the protein is changing shape doesn't mean that it's denatured. It may change shape and not work as well, or it may change shape and work better. Changing shape doesn't mean that it's denatured, so don't use those terms interchangeably. Another extreme condition that can denature a protein is the concentration of molecules that can hydrogen bond. And too high of a concentration of molecules that can hydrogen bond can denature a protein. And again, this is assuming that there's hydrogen bonding structures, attractions within that protein that keep it together. And just like the salt concentration, generally above a 10% weight by volume concentration of molecules that can hydrogen bond can denature a protein. And this is the reason why we can preserve foods in sugar. Sugars like glucose or sucrose, lots of hydroxy groups, lots of alcohol groups that can hydrogen bond, and that high of a concentration can make the proteins in those bacteria not work very well. Another variable that can interfere, environmental condition that can alter the structure of a protein, is carbon-based solvents. Remember, carbon-based solvents, these are hydrophobic molecules, and they interfere with hydrophobic interactions. And what's too high? Well, you don't have a lot of carbon-based solvents in your body, but if it went above about 2% weight per volume, that could denature proteins that have a lot of hydrophobic structures. If those of you that have ever washed your hands with laundry detergent, remember detergents are considered a carbon-based solvent, your hands start to hurt afterwards, and your hands may start cracking a little bit. There's structural formulas that keep your cells together. Structural proteins have a lot of hydrophobic forces, hydrophobic interactions that hold them together, and we'll see that in some upcoming slides. But if you add detergent, well, you're denaturing those structural proteins, interfering with those hydrophobic forces, and they don't work as well. They don't function as well. And I've done this. I've washed my hands, not recently, but I used to, with detergent after working with some grease or something like that. It really hurts your hands if you do it for a few days in a row. You denature too many of your structural proteins, and it can start hurting. Okay, what's another extreme condition that could denature a protein? Remember pH. pH can alter the conformation. And these extreme pHs are pHs less than 2 and greater than 12. And the fact that proteins get denatured at these two extreme pHs, it can happen to any protein whether there's salt bridges or not. And the reason why, which is kind of different than these other ones, is that at these two pH extremes, there's a high concentration of certain chemicals in there. When you're at a low pH, there's a high concentration of the hydronium ion. And remember, the hydronium ion, it can react and break a peptide bond. If you cleave one amino acid anywhere in that chain, that protein's not going to work anymore. So what I wrote right there, a high concentration of hydronium ion can break a peptide bond. And again, breaking any peptide bond can denature a protein. When the pH is greater than 12, there's a high concentration of the hydroxide ion. And even though we don't need to know this chemistry for this chapter, you learned it in organic chemistry, the hydroxide ion can react with an amide group and break a peptide bond. We don't find this extreme pH in our bodies. Remember, the highest pH is around 8 in your intestinal tract. But if you were in a potassium hydroxide or high hydroxide concentration, like in Drano, that's enough to break a peptide bond. Drano, just a little trivia, it's used to clear hair clogs in your drain. Hair cells are mostly proteins. Traditional Drano is a hydroxide salt. That hydroxide can react with the proteins in your hair, break them down into amino acids. All amino acids are quite water soluble, and you can clear that hair clog. And one last note about pHs. If a protein has salt bridges, pH changes will affect its structure. If there was a denaturing pH for a protein and we knew what it was, I would give it to you in that problem. And in the next chapter, when we go over enzymes, which are proteins, I'm going to give you denaturing pHs for those proteins. And I'll also give you the pH that it works best at. But we'll save those type of problems for next chapter. On the next 3 slides, we're going to look at the general classification of proteins. And proteins can be called simple, which means they're only made from amino acids. And you only need those amino acids for the protein to function. Proteins can also be conjugated, which means it needs other molecules to work. And a good example is the hemoglobin that we looked at earlier. That hemoglobin protein, there's 4 different chains, but you need the heme group in order for it to function. There's various classes of conjugated proteins, and hemoproteins would be proteins that contain heme group. And there's different types of heme molecules than the ones that we showed earlier. So for hemoglobin, you can have those 4 chains. You can have the polypeptides. Without the heme group, it doesn't function. So you can have the polypeptide and no function, no activity. If you add a group, like the heme group, and it attaches to the protein, it becomes an active protein, and it's called a conjugate protein. And a conjugate protein has biological function. So hemoglobin, even if those 4 chains were connected to each other, if there weren't the 4 heme molecules, you couldn't carry 4 oxygens per hemoglobin molecule. There's certain classifications of conjugated proteins, and it depends upon the chemical that's attached to the polypeptide. I don't expect you to be able to give this list. I just want you to understand that there's these types of classifications, and if I use these words, you should know that it's a conjugated protein. Certain proteins require metal cations, and good examples of cations that are used are iron and zinc. A lot of the various types of metals, which are all cations in your body, are associated with proteins. There could be carbohydrates associated with the proteins, and it's called a glycoprotein. There's lipids that could be attached to the protein and give it some function, and they're called lipoproteins. There's various types. There could be proteins that have phosphates attached, and they're phosphoproteins. Nucleoproteins have DNA or RNA attached to them. The next classification for proteins that we're going to discuss is type, and there's 3 main types. There's proteins that are considered fibrous, membrane proteins, and on the next page we'll look at ones that are considered globular proteins. Shown down here is a diagram of a typical fibrous protein. Fibrous proteins are usually made from 3 polypeptide chains. And that's what this diagram is trying to show here, 3 chains. These chains are long, straight chain molecules, and they're intertwined. This would be like having 3 Christmas tree lights intertwined in this sort of helical structure. Helical structure. Yeah, even though they're intertwined like this, there's a lot of alpha helic interactions. A lot of those structures within a polypeptide chain. The individual chains that are within this structure, there's secondary alpha helic structure, but they're also considered straight chain, which means the R groups, the side chains within a given chain, one chain, don't overlap and have any tertiary interaction. So it's straight chain, so there's no tertiary interaction structures within a fibrous protein. The fibrous proteins, they're said to be insoluble molecules. What does that mean exactly? It means these 3 chains are held together by hydrophobic interactions. The quaternary interactions is predominantly hydrophobic interactions. And this is especially true when the fibrous proteins are first synthesized. Later on, you start having bonding that can occur, like the disulfide linkages, that type of bonding between the chains, and it's called cross-linking. But definitely for this class, all I want you to know is that the quaternary interactions for fibrous proteins are hydrophobic, not hydrogen bonding. And that's the reason why we get the classification insoluble. No protein's soluble in water. It just means that water and the fact that it can hydrogen bond can't weaken the interaction between those chains. So the side chains, the R groups, must fall within that classification of hydrophobic nonpolar side chains. If you looked at a lot of the fibrous proteins that are in your body, proteins like collagen, they have a lot of their R groups that are in this category, neutral, nonpolar. And you'll see a few examples where glycine is a major component. So let's write that down here. The side chains, the R groups, must be neutral, nonpolar. So when you look at a protein, a fibrous protein, if I ask you a question like what type of interactions are present for primary, secondary, tertiary, or quaternary, well, it has all 3 except for 1. Every protein has primary, the sequence of amino acids. Fibrous proteins have a lot of alpha helic structure within their chain, so it definitely has secondary. Fibrous proteins are straight chain. There isn't, and you need to know this for the exam, there isn't tertiary. There's 3 chains, and any sort of interaction between any polypeptide chain is quaternary. There's quaternary interactions. Note, for the exam, I expect you to know this general information for a fibrous protein. In fact, when you do your practice problems, the problem itself assumes that you know this information. One, that there's 3 polypeptide chains that are held together by hydrophobic interactions, kind of like hemoglobin, but hemoglobin has 4 chains that are held together by hydrophobic interactions. And I also expect you to know that there's secondary structures. Remember, every secondary structure is hydrogen bonding. Okay, the next type of protein type is membrane proteins. In membrane proteins, where do you think you find those? Well, you find them in the membrane. We discussed cells a little bit, and we discussed the fact that every cell in your body has a membrane. But what does exactly mean? What's the composition of a membrane? So let me draw a circle to represent a cell. So there's the cell, and this would be the membrane. The membrane, it's very fluid, and it's very nonpolar. It's very hydrophobic. The membrane is like having a nonpolar hydrophobic solvent. It's kind of like the consistency of fat and oil. If a protein is part of the membrane, that protein, I'm going to draw it in red, it's long like a Christmas tree strand, and it may kind of zigzag in and out. There's portions of the membrane protein that are hydrophobic that would be somewhat dissolved, not really dissolved, but dispersed throughout the membrane. And there's parts that's extending out of and into the cell. The cellular fluids in here, it's water-based, and the fluids outside the cell is also water-based. It's very polar outside the cell and inside the cell, and the membrane is nonpolar. But membrane proteins, they're embedded in the membrane, and they serve some sort of function. These membranes could be an enzyme, they could be receptor molecules, they could be transport molecules. We'll see examples of all of those as we go through this course. The last type of classification for type of protein is globular. And globular proteins, they make up the most types of proteins that are in your body. You have thousands of different types of globular proteins. Structural proteins, fibrous proteins, make up the biggest mass of protein in your body, but globular makes up the most variety of types. Globular proteins are like our Christmas tree strand that's wrapped up into a ball. So these are very big molecules, and they form these little tiny particles, and these particles are dispersed throughout your aqueous environment. So here's kind of a diagram of a globular protein. What gives the protein structure in the interior is a lot of hydrophobic attractions that give it that ball-like particle structure. On the surface are a lot of R groups that are charged. So the surface is very hydrophilic, meaning the surface is very polar, meaning the surface is charged. Globular proteins definitely have a lot of tertiary hydrophobic interactions in the interior. There could be other tertiary interactions, but you would need to be given that information. Example proteins that we've already seen so far that are considered globular, well, one is insulin and the other is hemoglobin. And if we go back to those slides where they were given, we can kind of look at their three-dimensional structures. So here's insulin. Insulin, this globular protein, it's found in your blood moving by itself. It's a tiny particle. It's produced in your pancreas and attaches to protein receptors. But that very interior, there would be a lot of tertiary hydrophobic interactions giving it its structure. On the surface, there would be a lot of charged groups. Hemoglobin, and here's the structure of hemoglobin, and it's given slightly different without all the atoms. The ribbon shows the backbone. But it's a particle. This, though, instead of being freely flowing within your vascular system all by itself, it's found within the red blood cells that are flowing through your vascular system. But these tiny particles, they can move around and diffuse around within your red blood cells. They have a globular structure. Before we're done with this chapter, we'll see other examples when we list protein functions of other globular proteins, and membrane proteins, and fibrous proteins. On the next few slides, I'm going to list the various functions that proteins do in your body. There's 8 slides, there's 8 functions. I would make flashcards because listing these 8 functions is definitely something you're going to have to do on the exam. You're also going to have to give one example protein for each function. Your example protein doesn't have to be the one that I give on the slide, but you need to be able to give one. On all these slides, I'm going to give a lot of extra information. There's 8 slides. There's a lot of things that are said here. All I expect you to be able to do is to list the 8 functions and to give an example protein. Anything that happens in your body is the result of the functions of a variety of different types of proteins. Muscle movement, for example, that occurs because of a lot of different types of proteins and their functions. Okay, the first listing that we have on this slide, proteins, the catalysts that are in your body, those are proteins. Catalysts are also called enzymes. And on this page is an example enzyme, and it's called hexokinase. As you'll see in the next chapter called enzymes, anytime you see a word in this class and it ends in ACE, A-S-E, that's the name of a catalyst. That's the name of an enzyme. Hexokinases, they catalyze the reaction in which a phosphate is attached to a carbohydrate. 6-member carbohydrates like fructose and glucose. There are actually 4 different hexokinases in your body. The 4th one is called glucokinase, so that's number 4. And these structures down here are for glucokinase. All 4 of them are used in the catalysis of glucose reacting with ATP to form glucose 6-phosphate. And we've seen this reaction a few times, but I had to kind of make it curve down there to fit it on the page. Glucokinase, or any of these hexokinases, they're an enzyme, and you write the name of the enzyme above or below the arrow. It's not a reactant or product. These hexokinases are also metalloproteins, meaning they have some sort of metal cation that's associated with them. Magnesium, and we'll make it the correct charge, 2+, is a metal cation that's used in any ATP reaction in your body, including this one. Glucokinase, actually all the hexokinases, here's a new word, monomeric. Monomeric means it's a single chain. So there's no quaternary interactions in glucokinase or any of the hexokinases. Note, on the exam, I may describe a protein with some of this vocabulary. So monomeric means a single chain. Okay, just a few interesting things about this protein. You can tell by looking at this structure, there's some alpha helix type of structures. And you could be given the structure of an enzyme like that. Over here is a different diagram of the enzyme. This arrow pointing right here, this is where the reaction occurs on the enzyme. And you don't need to have that memorized, it's just an arrow that's pointing towards that. This protein, this glucokinase, changes shape based upon chemicals that are in here. And that shape change that can occur, well, look what's happening over here. That's with the shape change. That active site, it's like the door's closing. That change in the conformation of the active site means that this enzyme in this state right here isn't going to function very well. It would be hard for a reactant to get into this catalytic process when it's being closed like that. We'll see that a lot of enzymes, not every enzyme, but maybe 30% of the enzymes that are in your body, chemists say that they're controlled, meaning their shapes can change so that they work worse or better depending upon the environmental conditions around them. What's interesting, the control is mostly due to the molecules that your body produces or uses up, molecules that are part of your pathways. So as chemicals and their concentrations change, how well the proteins that are used to produce those molecules or use up those molecules, how well they work changes. Molecules and the concentrations in your body have control over those dams that open and close for pathways. We'll see that as we present more and more of the chemical pathways in upcoming chapters. But for this slide, all this extra information, just bear in mind, All you need to know is to be able to list that and to give hexokinase or glucokinase or any other enzyme that you know as an example protein. Another function for proteins in your body is transport. A lot of molecules that are insoluble in water, and your blood and your cellular fluids are full of water, have to be transported. And an example protein that transports on this page is serum albumin. Hemoglobin could be another example protein. And note, all the example proteins that I'm giving in this function section, most of them we'll see later when we look at our metabolic pathways. Serum albumin, this is found in your blood, just like hemoglobin's found in your blood, but hemoglobin's within a red blood cell. Serum albumin is like insulin. It's a single protein, it's a globular protein that's a tiny particle that's diffusing around in your vascular system. It's the one that's the largest concentration of proteins in your blood. It's produced in your liver. It has a lot of different functions, but one function is to transport molecules that are insoluble. And where we're going to see its usage later on in upcoming chapters is we're going to see that it transports fatty acids from cells called adipose cells to other cells. This protein, it's a monomer, it's a single chain. Its molecular weight is around 67,000 grams per mole. It has more functions than just transport, but the main example we're going to see for it is the transport of fatty acids. It has 10 binding sites for fatty acids. And you can see those labeled throughout in this picture. But again, for the exam, just be able to list transport and be able to list this one, serum albumin, or hemoglobin as an example protein. Another function, number 3 on the list, is storage, and an example protein is myoglobin. This myoglobin, its structure is shown down here. It has a heme group just like hemoglobin, and that's that heme group that's right there. This heme group is, the iron in there is iron 2+, and oxygen, O2, and its lone pairs combine to that heme group. Myoglobin is found in muscle cells, and its main purpose is to store oxygen to be used later by the muscle cells when needed. You find a much higher concentration of myoglobin in certain types of mammals. Those mammals hold their breath for a long time. Those mammals are creatures like sperm whales that dive deep into the sea. Those animals that hold their breath for a long time and their muscles are moving a lot, they still need oxygen. And the purpose of myoglobin is to make a large reservoir of oxygen when they're not breathing. Animals that dive deep, like seals or sperm whales, and hold their breath, express a large concentration of myoglobin per muscle cell. A couple notes about myoglobin. Myoglobin, this is what gives meat, the steak that you cook, that reddish color. When iron in this heme group, when it's 2+, it's red. For some reason, when you cook meat, it changes color, and it's because of the iron that's part of the heme group. When you cook meat, you expose it to high temperatures with oxygen. That oxygen in the air oxidizes the iron into iron 3+. And iron 3+, it's a brown color. So there's a conversational piece for your next cookout. Okay, another thing, when this reaction happens, the reaction is when the oxygen binds to the myoglobin, it's a lone pair that binds to it. There's other chemicals that you take into your body that have lone pairs. And something like carbon monoxide that has a lone pair, that can bind to the heme molecule. It could bind there, and it could prevent the oxygen from binding there. Note, that can also happen with the heme in your hemoglobin. And if that carbon monoxide is attached to the hemoglobin at the heme groups, it can't carry oxygen. Another molecule that some of us, and you shouldn't, take into your bodies is nitrate. And nitrate it has lone pairs too, and those lone pairs combine to your hemoglobin or your heme molecule in the myoglobin. You find lots of nitrates added to foods to preserve foods. If you're an athlete and you're going to compete and you're doing a quick sprint where you need your oxygen from your myoglobin, it wouldn't be a good idea to have a lot of nitrates from your beef jerky before or the day before a race. It means there'd be a lot of nitrate connected to your heme molecules and not oxygen. Note, cyanide also does the same thing. Cyanide has lone pairs and it combined to the heme molecules. Cyanide poisoning, it kills you because the cyanide binds to the heme molecules and prevents oxygen transport within cells. Another function is motion. So your muscles moving, your hand moving as you're taking notes. That's a chemical reaction, and it involves proteins. And it's involving proteins that are changing shape due to a chemical reaction. Good examples of proteins that are involved in muscle movement, there's proteins called actin and myosin. These molecules, there's lots of different types of myosins. There's like 40 different types that we know about that are in your body, and they're found in most cells, but they're involved in muscle movement or in any type of movement that you can think of that a cell does. When you have this movement, the actin, those proteins remain relatively stationary. It's the myosin that change shape, and it's due to a chemical reaction with ATP. But here we have kind of the shape of it. So this portion right here, that's the myosin. There's a chemical reaction that happens on the surface, and we'll describe it in a little more detail in a second. But there's a chemical reaction that happens about right there. So there's my arrow. And this portion of it changes shape, and it kind of cocks this way, upward. And then you have a reverse reaction, and it cocks down. So the head kind of flips up and then flips down, and that flipping makes it go around the actin portion. So that flipping will move this head to this side, and you get muscle movement. Over here, this is a diagram, a part of a diagram of a myosin protein. Myosins are pretty complicated. This portion down here, it kind of looks like a fibrous protein. Well, it is. Fibrous structures are part of the myosin. This portion up here is called the head. That's this portion that changes shape. Kind of the connecting part between the two is called the neck. If you look at the different colors in here, there's actually different chains that are part of the head structure. Not that you need to know this for this exam, but the reactive site is about right there, and ATP will react, and a phosphate is attached. You're attaching a negative charge. That big negative charge creates some repulsions, and you get a big conformational change. So what I wrote there, ATP reacts and attaches a phosphate, and that -2 charge that gets attached causes repulsion, and you get a big conformational change, meaning that head moves up. The phosphate is then removed, and it'll go back to its original position, and you get the movement. The net reaction from the myosin head moving from that position up to here is that ATP is used up. It's a complicated series of reactions, but here's the net reaction. ATP plus the two proteins forms ADP and a free phosphate and actomyosin. This is something you should know for your upcoming exams, especially exams 3 and 4. When you exercise and you do muscle movement, ATP is used up, and ATP's concentration would decrease as it's being used up. For exam number 1, you don't need to know this. Again, you'll need to know the protein functions and example protein, but when you start to exercise, ATP usage increases, and its concentration decreases. Another function for proteins is mechanical support. And an example protein is collagen, and there's lots of collagen in your body. As far as mass, it's about 25 to 35% of the mass of all the protein in your body. It's a fibrous protein, so there's 3 chains that makes up collagen. often, just in general, if you look at collagen, it has repeating units. And usually, like we mentioned earlier, one of the amino acids is glycine. And then there's some other non-polar amino acids, so X and Y. But often in collagens, and there's about 29 different types that we know about, you have repeating amino acid groups that are non-polar neutral, and one of them is glycine. Because remember, for fibrous proteins, you need to know that for the exam, the quaternary structure, it's hydrophobic. And all of these amino acid residues would be hydrophobic. And lastly, let's look at these three illustrations showing the protein structure of collagen. The first two are single strands of the fibrous protein. And this particular diagram, you can more easily see the alpha helical structure. Over here on the right is the 3-chain fibrous protein. And you can see the 3 different chains with the 3 different colors. Another function of proteins is your immune protection. The proteins that are involved in this are called antibodies. They're also called immunoglobulins. You can use any of those names that you want. Shown down here is an example of an antibody. There's lots of chains. There's 4 chains per protein. So when I say lots, that means 4. And you can see the different colors. These are glycoproteins, meaning there's sugars attached. The typical molar mass of an antibody is around 150,000 grams per mole. There's millions of different types. Most are very similar. Most have that molar mass. What makes them different is the tip area and the portion that has the carbohydrates. The carbohydrate, when it's synthesized, is very specific to the foreign body that's coming into your system. The foreign body is called an antigen. That antigen could be a molecule, it could be a microorganism like a virus or a bacteria, but the carbohydrate is synthesized to be complementary to that foreign body, to that antigen, and you get an attachment through intermolecular forces. And once, they call it tagged, once the antibody is attached, the immune system will work based upon that. So I just wrote a little information. Millions of different types of antibodies are created, and they're created when these foreign bodies come into your system. Most, again, are very similar. It's that carbohydrate portion, the glyco portion, that's very specific to the foreign body, the antigen. And that antigen, again, could be a molecule or it could be something bigger like a virus. That carbohydrate portion, that portion of the glycoprotein will attach to the antigen, and then there's other parts of your immune system that will get rid of that combined complex. But again, remember for this exam, you just need to know that listing and an example. And you can say antigen or immunoglobulin. Another function of proteins is control of growth, and a good example protein is growth hormone. This is a hormone that has a big influence on the reason why kids get taller. As a side note, this is a hormone that we're going to look at a lot this quarter because it also has a big influence on chemical pathways that we're going to be looking at. Like all hormones, there's usually multiple target cells that a hormone may have an effect on, and the effects are usually quite different. But we'll see that in upcoming material starting with exam number 3. The last of our list of functions is control of differentiation. And a good example protein or a class of proteins is called glucose transport proteins, abbreviated as GLUT. These are membrane proteins, and they're involved in the transport of glucose into cells or other monosaccharides into the cells. There's 13 different types of GLUTs that have been analyzed. One thing to note, our example protein is called glucose transport proteins, but it's under this category. Just remember, there's a category called transport, and we have serum albumin. So don't get those two mixed up, but it's just a naming thing. But if we look at the structure, here's a common structure of a GLUT. It's a helical structure, and here's the membrane from there to there. And the job of these transport proteins, these GLUTs, is to transport the monosaccharides like blood glucose from the blood into the cell. We'll learn some more specifics in upcoming chapters, and again, you don't need to know this for exam number 1, but GLUT1, this is found in every cell, and it transports glucose into the cell. GLUT4 is only found in a few cells like the liver and muscle, and it's hormonally controlled. We'll discuss what hormones later, but they're all a little different. GLUT5, which we'll see later, its job is to transport fructose, not glucose. So there's the list of protein functions that I expect you to be able to list, and you need to be able to list an example protein. And there's one thing that I hope you take to heart as you finish off this chapter, and I've said it a bunch of times and I'm going to say it again. So here's a statement I hope you take to heart. Protein function is based upon shape. So all these functions that we've described, everything that occurs in your body is based upon the protein shape. Okay, this GLUT, it doesn't chemically react. The fact that it can transport fructose or glucose has to do with its shape. And activity is, its function. Shape and activity change as concentrations of chemicals in your body changes. Chemicals in your body, these chemicals, these could be chemicals that are part of your metabolism. So these are chemicals that you produce and use up. Those chemicals that you produce and use up, as their concentrations change, the function of proteins in your body changes. And we'll see later, and as we've mentioned before, a major strategy of your metabolism is to keep concentrations of chemicals constant. Like you try and keep your cellular glucose concentration constant. If you're producing too much, those chemicals, those proteins that function to produce glucose, their activity will change. If you have too little glucose, the chemicals, those proteins that use up glucose that are part of those protein functions, they don't work as well. Also, which is just as important, the food molecules, the chemicals that you bring into your body have a big effect on the proteins. And you hear all the time in the medical field, what's your environment like? Well, it's talking about what chemicals you are or are not bringing into your body. Of course, some food molecules are part of your metabolism, like glucose, but there's a lot of molecules in foods that you eat that aren't part of your metabolism. They're just molecules that diffuse into your body and may affect the conformation and the function of proteins. And we'll give an example of some molecules that are found in broccoli in a little bit. Also, it doesn't have to be food molecules. There's other chemicals in the environment that you may bring into your body that can have an effect on the function of proteins in your body. Like lead the cation, and we'll give examples of its influence a little bit later, or maybe cyanide, or maybe aluminum the cation that you may get from cooking with aluminum. But anyway, the whole point is external molecules that may not be part of your metabolism can also have an influence on protein function. But the main thing we're looking at this quarter as we go through this entire quarter, molecules that are part of your metabolism, the concentrations of those molecules have an extremely big influence on the rates of chemical pathways in your body. It's pretty fascinating, and it's an amazingly complicated yet simple control scheme. For the remaining part of this chapter, we're going to give an overview of protein synthesis starting with your DNA. Later on this quarter, we'll look at the chemistry that's involved in the process. And we're going to give an emphasis on your type of cell, so not bacteria. So let's look at human cells. In the human cell, there's 46 chromosomes. Every cell has the same chromosomes. What exactly is a chromosome? This is what contains the DNA molecules. And these are huge, huge molecules. But each chromosome contains 2 DNA molecules. And they're held together through hydrogen bonding intermolecular forces. There's also protein molecules that are bound to the DNA molecules. And these protein molecules are called histones. So if we look at our diagrams down here, this ribbon, this is representing two chains of DNA, two DNA molecules, and there would be two per chromosome. Remember we gave ATP, adenine, guanine, those same bases and the ribose and the phosphate are all part of the structure. Between the two molecules, these dashed lines here represent hydrogen bonding. And the base molecules like adenine, guanine, they're complementary shapes so that they fit together. The two molecules, if you go up and down the two molecule chains, there's complementary bases, which are the adenine and the guanine molecules, that hydrogen bond to each other from one molecule to another. These chains of DNA form a helical structure, and it's called a double helix because there's two molecules forming that helical structure versus one chain in a protein forming an alpha helix structure. Wrapped within these DNA molecules are proteins, again, that are called histones. And these histones, they have different functions. One of them is to add support. So proteins are big, DNA molecules are enormously big. So hundreds of millions of grams per mole per DNA molecule. They're huge. If you look at a complete chromosome, you can actually see it. Not with your plain eyes, but using optical microscopes. And here's a chromosome containing two DNA molecules. Okay, these histones, they also serve a function for gene expression. Proteins are made from genes, which are a sequence of bases that you find in a DNA molecule. That's a gene. When the two DNA molecules are attached, the gene is not expressed. So what I wrote right here, the gene is not expressed when both strands are attached to each other, meaning you have to break the hydrogen bonding between the base pairs in order to express that gene. These histones can play a role in that. They can change shape to make it more easily or more likely that those strands can be separated from each other. So what I wrote right here, that histones play a role in gene expression. And we'll learn what gene expression means, but when a gene is expressed, it means a protein could be most likely synthesized. RNA and DNA are huge polymers, and they have monomers. The monomers are called nucleotides. And within that nucleotide are a few molecules. One of them is phosphate. Another one is a sugar, and it could be ribose or 2-oxyribose, which one's used for RNA and the other one is used for DNA. What makes the monomers different, and there's 4 monomers per RNA type of polymer and 4 monomers per DNA polymer, is the nitrogen bases that are used. So shown here, image I just blew up, would be four monomers connected together. So a monomer would have phosphate, so there's a phosphate. This one has ribose, and what makes each monomer different is the base. So one type of base would be adenine, another type would be cytosine, there's guanine, and thymine. This portion of it looks pretty similar to our ATP molecules and our coenzymes. But this is, showing the construction of our bigger DNA and RNA molecules. Note, for DNA and RNA, there's one different base, but we'll talk about that a little bit later. If you look at the nucleotide residues, each nucleotide, the molecular weight is slightly different, but the average molecular weight of a nucleotide is around 500 grams per mole. If you look at the different DNA molecules that are in your cell, they can range from having 1 million nucleotides to 100 million. Times that by 500, it gives you the molecular weight of a DNA molecule. They can be huge, huge molecules. As far as your genetic information, the order of the monomers that you find in the RNA and DNA molecules, well, in particular DNA molecules, that order of bases that you see in your DNA, that's your gene. And that order is related to the order of how amino acids are sequenced in a protein. Later in the quarter, we'll look at that chemical process to go from your DNA to a protein. For this chapter, again, we're just going to be looking at some general information. And that general information is just to show you how you can go from the DNA molecules to making proteins. Okay, so the genetic information, the order of bases in your DNA, that order is used to make RNA molecule. That process is called transcription. So in your nucleus, you find the DNA molecules. In the nucleus, you make RNA molecules. And those RNA molecules, well, there's monomers too, and there's bases. The order of bases in your DNA sets the order of bases that are in your RNA molecules. That messenger RNA is made from a gene that's in your DNA. This messenger RNA will somehow go through the membrane of the nucleus and go into the cytosol. It's in the cytosol where you have protein synthesis. And here's the complexity, a little bit of complexity. There's a base structure called a ribosome that the messenger RNA attaches to, and there's enzymes and stuff involved in the process. But once this messenger RNA is attached to the ribosome, protein synthesis starts. And protein synthesis is called translation. There's other RNA molecules that are always present in your cytosol, and they're called transfer RNA. And these transfer RNAs, each one takes a specific amino acid to the growing protein chain. The transfer RNAs also have, they're made from nucleotides, and they have certain bases, and they have complementary bases that can hydrogen bond to the messenger RNA. So, very specific transfer RNAs with very specific amino acids will bind to the specific order of a messenger RNA, which came from the DNA, which makes a very specific order in which amino acids are put together here, and there's an enzyme that catalyzes the formation of that peptide bond. That's pretty amazing. So the exact order of the DNA makes an exact order of an RNA. The transfer RNAs place very specific amino acids with that order. This is why your body is so good most of the time at putting amino acids in the correct order to make a protein. And right here we're showing the growing chain of the polypeptide. Also remember what we said earlier, the structures of the protein start to arise as the protein is being synthesized. And we can see this in the next diagram on the next page. So here's the diagram, and this is representing the growing chain. As the chain is growing, you start to get the structures, they start to form. And it says just secondary structures, tertiary and secondary structures, so we'll put an and, start to form when you have those attractions and those groups are next to each other. Okay, one last comment, and this has to do with control. So control, what does that mean? You have gene expression, you make proteins. You don't want an endless supply of proteins in a cell. So you want to control how much protein is being produced. And it's amazingly complex, the control that happens for protein synthesis. And every aspect of protein synthesis, starting with the DNA, we have examples of control. For some genes, there's a modification of the DNA, a chemical modification, so that the gene can start being expressed. Sometimes genes are always open, so those two strands are away from each other at that gene, and they're always expressed. And sometimes it requires a chemical reaction. Okay, the next part was making the messenger RNA, so the synthesis of RNA. That's transcription. And this involves enzymes. And those enzymes are proteins. And those proteins can change shape, and how well they work can be controlled. The next one we didn't talk about on the previous page, but sometimes messenger RNAs, they're synthesized, but they're synthesized in an inactive form, and they have to be modified so they become active. And this is called post-transcriptional. That's the process, and there can be control. There's enzymes, there's proteins involved in that process. Translational, again, that's the process of synthesizing the protein down here. There's enzymes involved in that process, and there could be control with those enzymes. Post-translational, after the polypeptide is made, some polypeptides are made in an inactive form, and they have to be modified to become active. And a good example are digestive proteins that are secreted in your intestinal tract. They're stored in the cell, and they're secreted when you eat food. If it's a digestive protein for a protein, you don't want it to be active inside the cell. You only want it to become active when it goes outside the cell into your intestinal tract. But post-translational, proteins are modified to become active. And usually modified means there's some sort of chemical reaction, like part of the polypeptide is cleaved or a phosphate is attached, something like that. As far as studies on our genome, we certainly know far less than what we know on control, but there are plenty of examples of all of these controls throughout for different types of proteins. A lot of studies in general, instead of looking exactly how things are controlled, they just know that if you change this environmental condition, there is some control. And when I say control, that means some genes are expressed more when you change this condition, meaning the synthesis of proteins of that particular protein increases. If the control is the opposite way, it means the genes are expressed less and there's a lower rate of synthesizing that protein. And a good example I'd like to get is a study that was done on males with early term prostate cancer. For all these males, they introduced 1 serving of broccoli 5 times a week. Previously, none of these males ate broccoli. There are certain chemicals in broccoli that aren't part of your metabolism, but they were known to influence gene regulation. These scientists they got a baseline concentrations of different proteins in the prostate cells. So again, protein concentration is related to gene expression. What they found was that 400 genes, the expression of 400 genes changed, meaning the concentrations of 400 proteins changed. Some of them increased in concentration, some of them decreased in concentration when these males started eating broccoli. These chemicals in the broccoli, we're not exactly sure what they influence as part of these, what step of the pathway it had an influence on. It could have been right at the gene with those histones, those proteins, or maybe somewhere downstream. All they know is that environmental change of introducing broccoli into their diet had an effect on the synthesis, on the rate of synthesis of over 400 proteins. These chemicals that were in broccoli, they didn't chemically react with proteins that are part of the genome and genome expression, but somehow those chemicals influence the shape and the function of different proteins.